under orientation of Dr. Patrick Hogan, also at LJI. And we further characterize the mechanism of action of one heat selected during this high throughput screen. So a balance of my postdoc, my experience with a postdoc abroad. The US has way more resources than Brazil, what enables good quality research. Reagents here arrive in 2 days, so besides having money, it's faster. But I was thrilled by recognizing how we Brazilians are good at researchers. It was a big load of knowledge and new technologies to learn, but we have the same capabilities and we are well trained to perform experiments as any other scientists, such as Americans or Chinese. And Brazilians are in general very well seen by PIs here, especially here, like my experiences in San Diego. But I have to admit that I will be accepted to this lab by myself with my CV. So when you apply to labs abroad, you compete with scientists that already have publications in high impact journals, usually from countries that have more money, so they have more money for the research. So I'm really thankful for João for opening the door. Of course, I proved myself later, and my PIs here in the US start to pay me. But this is a big advice I have to you. Establish good relationships. Take time to develop these relationships. If I could go back, especially during my time at LJI, I would have taken more time to build relationships with other scientists and other PIs to further have more recommendations. Connections and indications matter a lot, especially if you are going to biotech. So I was in my postdoc and I felt overwhelmed by the life, I would say like every single PI I know has. I felt a lot to imagine myself writing grants, supervising people, taking care of money, teaching, revising papers. Since English is my second language, I find more challenging to express myself and to really write well. I'm very a detailed person, so it takes too much time. And like, I was really confused if like I really wanted to become a PI. So San Diego has a big biotech industry sector, and I was always curious in how it would be to work in biotech. So my downside at that point was I didn't have a green card, and biotechs usually don't sponsor visas, different from academic institutions. And I've heard from a lot of people that I will not get a job in biotech because of that. But I decided to apply, and after some months, I got a job. So I got an offer from ASEA Therapeutics. Hey, just a minute, because I'm not seeing you. Are you still hearing me?
Yes.
Oh, okay. Just to make sure, because I'm not seeing you. Sorry. Okay. So I had this job at ASEA Therapeutics as scientist in immunology. It was a small startup working with new immune oncology targets. And it was the first time that I worked with human samples, and I simply loved it. So I love working with human samples. That is what I always wanted. I love the research. I love the environment of a startup. You have to be proactive, multitask, fast-paced, and this is very me. I had a better work-life balance, not working like late hours during the days, not working during weekends. And salary is also like way better than academia. After some months working at this company, we submitted a provisional patent application. So we were super thrilled about that. Our research was going super well. And from week to the other, the company shut off. Like they closed the company. I was working there for almost 1 year. It seems there was a mistake in a clinical trial that the company had in China that didn't have anything to do with our lab. But the entire team was laid off. And then it was like, oh my God, what do I do now? So at this point, my ex-boss, Heini, was really pivotal for my next transition. So he revised the entire team's CVs. He helped with references. In a month, I was found by a headhunter through LinkedIn, and I got an interview, and then I got an offer in another biotech called ClearNote Health, also as a scientist in immunology. So this company was working with human blood from patients with cancer, and they wanted to evaluate if immune cells from blood would improve the cancer detection. So I set up the entire immunology lab. I worked with them for 3 years. At some point, I was promoted to senior scientist. In the beginning of 2023, we also fill up this patent application that shows how the Buffy code, the genomic DNA of Buffy code, can improve the cancer detection. And we had a paper ready for submission, and then I was laid off. So what happened was the company didn't find investments, so they couldn't secure funds for the next series of investments, and they had to close all the side projects. So now the company is still is in place, and they have a test to detect pancreatic cancer that is approved by FDA, and they are in clinical trial. But I was not involved in this project. This project didn't have anything to do with immunology. Was barely was just genomic DNA from cell-free DNA. And then I was like, okay, you have to look for work again. A month later, I got my green card, and then I thought, okay, now it will be easy to find a job because I don't require a visa. But it was not. This was May of last year of 2023. So 2023 was a year of big layoffs in biotechs. I still have a lot of friends that don't have a full-time job since last December. The market is slow in biotechs right now. So it seems that during COVID, a lot of investments went to startups, and 3 years later, several of them didn't develop a product, so they could not secure funds, and this decreased their market value. So I then, looking for jobs, I applied for a position at the Moores Cancer Center that is part of UCSD. And is in a lab that works, they focus more in myeloid cells. So my entire career, I worked with T cells. And when I was in biotech, I really miss having more knowledge in myeloid cells, because the signal we found in the Buffy code to detect cancer in the previous company came from myeloid cells. So during the interview, I've learned that I will be able to work with ex vivo cultures of primary human cancer tissues and with new cancer therapeutics. So I accepted the job, and then I'm back in academia as a senior scientist. When I started in this lab, we were working just with lung, and now we expanded to renal and to head and neck cancer. We are doing multiplex immunohistochemistry, single cell RNA-seq, spectral flow cytometry, and we are expanding a lot. So today I'm coordinating this project. There is the immunoprofiling of tumor microenvironment in human tumors. And it is a research that brings me fulfillment, and again, the opportunity to expand my knowledge and to work in a subject that I always wanted. So the same way that I was told before that I will not find a job in biotech because I didn't have a green card, I was also told that once you leave academia from biotech, there is no way back. There is a gap in terms of publication in my CV right now that I have to fill out to be able to be competitive and to get grants. And for sure, if I had taken longer than this 4 years that I work in biotech, it will be harder to fill out. But the message here is, I think that there are always a way to change sectors if you can bring value for the company. So how I see the future of my career, first of all, I'm open-minded and I'm super willing to take risks. I think the priorities change with time, and we should be okay in accepting that. A major change for me was when I became a mom. Our free time changes a lot when you are a parent. But I'm really open-minded for this. I told you before that one of my big concerns of becoming a PI was writing grants. So what I'm doing is I'm taking the opportunity to go for a professor track with the Moores Cancer Center, and I will attend a grant writing course with UCSD. So, and this will be next year. So after that, I will revise my opportunities and my strengths and take the next step. I've learned that stability is something that doesn't exist here, and I'm finally in peace with this statement. And I think that, like, in general, you need to be good. The competition is gigantic in both settings, in academia or in biotech. But I think that the recipe is to be prepared. Keep studying, getting up to date with literature, with new technologies, and really be aware of opportunities, because a lot of opportunities exist here. And to finish, I would like you to take some minutes to reflect on what defines success for you. What does it mean to be successful? And I want you to consider in this definition the ideal work-life balance that you would like to have. And I would suggest you to talk about this with people that you consider successful in a specific setting, to really learn about their lives and put yourself in the position and reevaluate if that's actually what you want for your life. The other question is, what are you really good at? Sometimes you want to be a professor and have a lab, but this demands like having great ideas to secure projects, secure funds, writing papers, teaching people. And of course, you can be trained, and this is part of our formation as PhDs. But do you really want to keep doing that throughout your life, throughout your career? Perhaps you are better troubleshooting, or perhaps talking about science instead of performing science. So I think you need to really realize what fits you better. The other point is risks versus money. So in my case, for example, I think that biotech has more risks, because especially small startups, they can close, as it happened to me, like from night to day. But because they are more risky, they pay way more. So are you willing to take risks? Are you emotionally mature to be laid off and perhaps having to look for jobs for months? Because that's the reality of the sector. On the other hand, like, you can be in a startup that after will be public, and you can make a lot of money selling your stocks, or not, like, or you prefer consistency. And I think it's good to think about that before making a decision. And I can strengthen more the power that connections have. There are positions that are not in the market. They are filled by recommendations. If there are 100 candidates applying for a position, and this is happening a lot in San Diego since last year, actually more than 200 candidates applying for the same position. And if you have a referral, the chances of at least you going for a on-site interview are higher. So try always to establish good connections. And if you leave your positions, like, try to live in good terms. I think this is pivotal. So in your path, don't compare yourself with the others. I think we always have different stories and different opportunities. And this is not to be mean, but I think that this touched me. Like, you are not a plant. If you're not happy, move. Usually you are not able to move as soon as you decide to move, but have a plan, talk to people, and make the arrangements needed for that. Don't be afraid to follow your heart. We spend a big part of our lives working, and life is too short to be unhappy and to be in a place that doesn't bring you fulfillment. So this is the message I have for you today. I am really thankful for all my mentors, for my family, my friends, and for myself. I think I've always believed in myself, and this is pivotal to achieve your goals. I think I have a great quality of life here. I believe the research I do is relevant, and it fulfills me a lot. And I would like to share this with you, these pictures that occupy a majority of my days and bring me joy. The right here is the Moores Cancer Center where I work. And again, here is my family, my husband and my son. And if my story resonates with you and you believe I could help in any ways, feel free to reach out. Here is my email. And thank you for hearing, and I'm happy to take questions.
All right, Gil, it was really great. Thank you. Thank you for sharing. Can you hear me?
Yes.
Okay, oh, good.
I'm
sorry for all the trouble starting up.
yeah.
I'm very sorry for everything, but now we are finally with everything working as should. Wondering if anybody in the audience here would like to ask a question, make a comment, anything. Just before they get over their shyness, I just would like you to talk a little bit more about those moments that you were laid off. I think especially the first one, the surprise of it, and how did you feel that the world was ending? It was an opportunity. How did you react to this new situation for you? Right, you had never been laid off before, right?
Before, yeah, no,
I can imagine.
Yeah, it was really tough. It was really tough, and I was in a visa here, so. When you are in a work visa in US and you are laid off, if you don't find another job in 30 days, you need to be out of the country. So it was really a moment when I was like, okay, so maybe I should go back to Brazil, or what do I do? But I think that like at that point I would say it's on the top more like strong moment. As I said, the research was doing well. Our research was doing well. Our team was doing well. And it was like in a Friday that everybody started to be tense and nobody understood what was happening. And like on Monday, the entire team was laid off. So it was really stressful. And it was really like, what do I do now? Why did I do this? Like, why did I move from academia? This would not have happened if I was in academia. But I think that it was pivotal, the support we had from our boss, like from Heine at that point. He was like, let's go the entire team for a lunch. I will revise all of your CVs. I will work with you for the next step. And it was really like a comfort. And as I said, like at that point for 1 month, looking for a job was my full-time job. I was in LinkedIn like 10 hours a day. And I was fine. I was found by a headhunter. Was not true recommendation at that point. But I think that this happens, and this is something that I need to be mature to understand that there is a biotech really fluctuates with the economy. So I joined biotech when like everybody was being hired and like salaries were high. It was super easy to get a position. My second job was the same. As I said, in 1 month I got a job. But since last year, it's down. So I have friends from Pfizer, from Takeda, from small biotechs that were laid off, and they are not like finding a job until now. So it's really like a wave, and it really requires emotional maturity to like do this, go through that.
Right. Thank you. And I think one last point that people would love to hear about. How was it returning to academia? How did you feel welcome, or people would just look at you? Oh, you went for the big money. Now you are back competing for our jobs. How this worked?
I think that back to academia was way easier than being laid off. Let's point like this. No, I think that when my current boss read my CV and when I went to on-site interview, she already told me like, you are my top candidate. So I did not have any problem. I was well accepted by the team, and it's really great. Like, it is a cancer research as it was in Rio, but here in San Diego. And they are very well connected. There is a lot of collaborations. We have a lot of collaborations with clinicians that do clinical trials. And our lab is doing like this top technology. So everybody is interested in collaborating with us. I didn't have any problem. Besides taking a pay cut. Like, of course, I took a pay cut going back to academia, but I felt very welcome, and the transition was easy in terms of research. And I think that, like, the way the biotech thinks, like this thing of being more focused and being more fast and being really like, you have to do connections, you have to be fast paced. I was always like that, and I improved these skills a lot. So I think it's helping in my current job now.
All right. Thank you very much, Gio. I don't think that we have any other comments here. So, actually, yes. We do have a question from Jonathan Arm. That is one of our speakers as well today.
Biotech, the pharmaceutical industry can have a bit of a bad rap for the.
I'm not hearing him.
Of the research.
Tá me ouvindo? Alô? Tá.
Yes.
Some magic here.
Yeah, I don't know.
I don't have Bruno's touch. Can you hear me?
Yes.
Okay. So sometimes the research done in biotech and the pharmaceutical industry is looked down upon a little bit, and sometimes doesn't see the light of day also for various reasons. Do you want to comment on the quality of science in biotech in your experience versus academia?
Sure. I've heard this a lot. Like, I think that this is like I hear this a lot from people from academia, but I don't agree with that position. I think it depends a lot of the biotech we're working with. Like like the first one that I worked, it was from 1 year, like 11 months to be honest. And we were developing new immunotherapeutics, and it was very similar to the work that I was doing during my postdoc. It was really well controlled. It was more focused because, like, everybody works together, and I'm more a collaborated person. Like, I like collaboration instead of competition. I think we can compete with the rest of the world. You don't need to compete inside your institution, and this is something that I really liked also in biotech. The entire group works for a specific goal. But the second company that I worked for 3 years, I was really impressed by them. And actually we published like a Nature Communication article during the time that I was working with them. So they work with 5-HMC methylations in cell-free DNA, and we were using immune cells because we were getting blood to improve the detection, and we got this. So I think that it was great, the research. I think that what is bad sometimes in biotech is decision makers. Like, who is the CEO of the company that makes the decision? Because in both companies that I work, it was a mistake there at the high level position. Like, there was a mistake in the clinical trial. They changed the protocol of the clinical trial, the clinical trial in the middle of the clinical trial. And like, when the Chinese FDA realized that, they asked for more 2 years of clinical trial, and this is a ton of money. So this should never have happened. And like, while I was working in the second company, it was a mistake too. I was in maternity leave, and they changed the method to isolate DNA that was super simple. Instead of being column-based, they start to use magnetic beads. And okay, it looked fine, but the idea was, okay, let's sequence some plates, and we spent more than $500,000 doing this. Instead of sequencing some plates and analyzing, trying to build a model with this new technology, because they were desperate for money, they were like, let's go for 1,000 samples. And they had an issue with this new isolation because like some C-rich areas changed during this isolation. And it was just like a naive mistake.
All right. Thank you very much. Thank you for being so candid about it, and it's really nice to hear from someone that has this really great experience. Right? It was good for your advancement as a researcher. It was a good experience, although you are back in academia. Would you consider going back to a biotech company if you got proper offer?
I think it depends a lot. I'm more mature now in terms of like where I will go, and I don't require a visa anymore. So I think I can choose more. Before I was just like, okay, well, who can accept me because I'm in this condition? Now it's just like I can choose. I think that like it will depend on how I feel writing the grants. But if I really don't like this, I will consider going back.
All right. Yeah. Great. Thank you very much. It was really, really great. Thank you.
Thank you. And feel free to reach out.
All right. Thanks. So we are going to move on to Dr. Jonathan Ahn, that has a little bit more experience than Juliana. He's been around for a little bit longer. I don't know where you want to talk from there. No, that's okay.
All right. Does this work? Great. Fantastic. All right. Well, I'll talk to the audience, but it'll turn my back to the screen, unfortunately. Okay.
That's okay.
So thank you, Bruno, for inviting me. So I started my career as a physician. I joke that I became a physician because my mother told me I would be a physician. There's some deep psychopathology in there somewhere. But I chose medicine, and that was going to be my career. My medical school was sponsored by the Royal Air Force. My father had been in the Air Force, and it seemed like a good way of getting a stipend as a student. Then I had to pay them back with 5 years of practical medicine. So I trained in the United Kingdom. I did my house jobs there. And in England, you have to do 6 months of medicine and 6 months of surgery. And I was going to be a surgeon until I did my surgical house job. And I realized that medicine was much more interesting, or at least it was to me. I appreciate surgeons would not agree. So I guess if I'm thinking about career thoughts, don't be afraid to change your mind. And it was a very simple decision for me. So I went into internal medicine. The Air Force trained me in internal medicine. And my next decision point was whether to stay in the Air Force, which would have given me an excellent career, an automatic promotion up until my late 30s. I would have become a consultant and left at the rank of lieutenant colonel or whatever that's worth. And it would have been an excellent experience. But I couldn't see myself staying beyond that point. Again, I did not think it would be challenging enough to just, if that was all that there was in my life. So I chose to leave the Air Force at a time when I had trained in general medicine, and now I needed to do subspecialty training. I had a couple of years of training in renal medicine, kidney medicine, but I wanted to switch to pulmonary medicine, studying lungs and inflammation. And I got very lucky interviewing with somebody who had 2 years of research funding to do a study for a pharmaceutical company. And in those days, you could do research for a pharmaceutical company. They paid your salary for 2 years, and you probably spent 1 day a week on their research, and you had 6 more days a week left to work on other research projects. And Naomi Eiser was extremely generous and gave my services to a chap called Tak Lee, who had just returned from Boston, where he had worked in the lab of someone called Frank Austin, whom I'll talk about a little more later. So I went into research because I had to. I was not going to get the next job unless I did research. Now, I was always interested in research. I had published some clinical research doing the renal job when I was in the Air Force. I had specifically tried to get to the renal unit in the Air Force because they did research. but it was very clinical research. So then I was very fortunate. I was going to do 2 years of research with TAC at Guy's Hospital in London. And he was very interested in leukotrienes before we knew that leukotrienes had a role in asthma. It's that long ago. And he was also interested in whether we could modulate leukotriene generation by feeding people fish oil. And as you may know, the fatty acids in fish oil get metabolized to a different set of leukotrienes and prostaglandins. And those leukotrienes have less inflammatory effect. This was an extraordinary research opportunity looking back on it. I was working with patients with asthma. We wanted to know what happened to their asthma when we gave them fish oil. So it was an early introduction to interventional studies. I had to work with patients to work out how much histamine it would take to cause bronchoconstriction, learning to do what we call bronchial provocation tests. So again, working with patients, but also we're feeding people fish oil. We're taking cells from people, taking them to the lab, and having a look at what the impact of fish oil is on their cells. What is the impact on their leukotriene generation, chemotaxis, expression of activating receptors? So not only am I learning to work with patients, I'm learning to work in the lab and do flow cytometry, isolating neutrophils and monocytes, running HPLC. I think it was an extraordinary opportunity, and I didn't appreciate it at the time, but I learned a lot of different techniques, and it taught me not to be afraid that if I needed a technique to answer a question, just go and learn it from somebody who has it, and not to be restricted. So that was a very productive time. My 2 years turned into 3.5 years, but we published a lot. And it was, just going back to something said earlier, it was a very collaborative group. We helped each other a lot with our experiments. And again, that it doesn't just get you co-authorship on a paper, which is helpful, but it also means that each of you is more productive. And I found that to be very rewarding. But then I had a decision to make. I went into this in order to be able to do the next job as a physician. And was I going to go back to my next stage of medical training to become a pulmonary specialist, or was I going to stay in research? So I applied for a job as what we call in England a senior registrar, the next phase of training at Guy's Hospital. And the night before the exam, I withdrew my application, which made some people quite angry because I was the internal candidate and I had a good chance of getting the job. But I looked at my opportunities. I was enjoying research immensely, much more than I had expected. I found it very challenging. Not that medicine is not challenging, but a very different challenge. And the other interesting thing, talking about work-life balance, is that if I was to go into back into the senior registrar job, every 2 or 3 nights I would be on call. Every 2 or 3 weekends I'd be on call. I had no control over that. Now, research, I would not have those constraints. The downside, as you know from research, is you can spend 24 hours a day, 7 days a week working, and there's still more to do. So you have to regulate that yourself. But at the same time, if I needed time out to take the kids to school while my wife went to her job, and we could alternate on those things, I just had much more flexibility to make our family life work. I think I worked just as hard in research as in medicine, but it just gave me much more flexibility, and I think that also played into the decision. So I was looking for a postdoc and ended up going to Boston to work in the lab of Frank Austin, which is where Bruno and I met. That was a tough training. Sometime you should ask Bruno to show you the list of sayings from Frank Austin. There's 22 of them. They include things like don't take vacations. If you slept last night, you had time to do the experiment. But it was a very good training. And while I was there, I learned some more techniques, molecular biology techniques in the days of wet lab cloning. So we purified a protein from the surface of mast cells, and we got the N-terminal amino acid sequence, and then used redundant oligos to screen a phage library. Took a long time to get there, the best part of the 2 years. We took a few wrong turnings on the way. But we succeeded, and we pulled out what turned out to be what is now the first member of what is now called the leukocyte immunoglobulin-like receptors, and would now be called LIR, leukocyte immunoglobulin-like receptor B4. So after an abortive 2 years back in London, I was on a J2 visa in the US. I had to go back to the UK for 2 years, but the family missed America. I missed America. My wife had 8 years of her childhood in Canada and the United States. And so there was a lot about the culture in America that gelled for her much more than the culture in England. So I was fortunate to have the opportunity to go back to Frank's lab as faculty. And then I set up my career as a physician scientist. I was at that point in my career, I needed grants wherever I was, whether it was going to be England or in America. So I went back. Was fortunate to get funding from the Burroughs Wellcome Foundation as a junior investigator. Got funding from the National Institutes of Health for funding, and then just built a research portfolio working again on the leukocyte immunoglobulin-like receptors, switching to the human counterparts, and working on what regulated leukotriene and prostaglandin generation in mouse mast cells. And could we, as mast cells shifted their phenotype, what was regulating the shift in generation of leukotrienes and prostaglandins, which enzymes were being implicated in that, and which enzymes were controlling the reactions. And that led us all the way back down to, or upstream, to phospholipase A2, that some of you are familiar with. When I started working on this, there were 3 phospholipase A2 enzymes described in humans. There are now, I don't know, probably 20. And we saw this huge explosion as people moved from wet lab cloning to in silico cloning. And we were looking at what was regulating leukotriene generation in the mast cell. And we made a bet on one of the particular phospholipase A2 molecules called Group 5. So we worked with Laurie Glimcher's lab, actually Mike Grosby, who was in Laurie's lab. Bruno worked to make the construct to get a knockout, and then Mike generated did the work with the ES cells. We got chimeric mice, and we were very fortunate it went germline, and hence we got the knockout. And fascinatingly, and this is always a fun thing in science, the knockout had a phenotype we did not expect. It had some loss of leukotriene and prostaglandin generation, but most intriguingly, I had a problem with phagocytosis, and that's what we pursued. Well, all this time, I had started working with humans and started working on very translational questions. What is the role of leukotrienes in asthma? And here I am now, I'm working with cells, I've got a knockout mouse, and I'm not thinking of a single way I can translate any of this into therapeutic opportunities in humans. The leukocyte immunoglobulin receptors, I didn't have the reagents and perhaps not the imagination to think what the next translational steps were. So I was getting rather frustrated. Some of my early work had been on a condition called aspirin exacerbated asthma, where people who take non-steroidal anti-inflammatory drugs get catastrophic asthma. And I tried to work on that in Frank's lab, and I presented to Frank some early, fascinating data on the ability of nasal polyps to metabolize arachidonic acid. We had a clear distinction between people who were aspirin sensitive and those who were not. Frank looked at the data, and he looked at me and he said, "You will never be funded to work on this. Cease and desist." So the boss told me not to work on it. I ignored him, but I couldn't persuade my postdoc to take the same approach. However, when I left some 10 years later, we were able to use the data I had generated to get funding for the next person. So he was wrong, I was right. But it was an interesting lesson for me. Sometimes the right experiment to do is the experiment your boss tells you not to do. Careful who you tell that to. So I was frustrated. I wanted to do something translational. I had a look at the pharmaceutical industry before, and I thought, I could not think of anything more boring than conducting clinical trials with established drugs. That I can see the attraction perhaps now, but then I did not see it. But someone asked me to go and interview at Novartis for a job in what they call translational medicine. How do you get a medicine that's been developed in the lab into humans for the first time? How do you know it's hitting the target? And I went to interview and I saw the work they were doing, and I came away so excited. I hadn't been that excited about science in a very long time. And it was just obvious to me this is where I should be. It was just, for me, the perfect place for a physician scientist. I would go to work in Boston, and if I'm seeing patients in the clinic, it's one part of my brain working. And if I'm in the lab, it's a different part of my brain working. And I was not bringing the two together. But at Novartis, that's exactly what we were doing. We were taking the basic research that was used to identify the relevant biology in disease and in animal models, developing medicines, and then taking them into humans. And it was, for me, it was just the perfect place to be. So I got to work on not just, my favorite molecule, phospholipase A2, or leukocyte immunoglobulin-like receptors. It was whatever was relevant to get to where we wanted to go. The other thing I like about working in a pharmaceutical company is, at the end of the day, either you develop a medicine that impacts people's health or you don't. It's not about just getting the next grant and the next paper, as valuable as that is, and as much as I value basic science for its own sake. But for me, it was much more easy to have a tangible end, tangible goal at the end. So I like that also. And again, coming back to teamwork, it's impossible to do that job without working with a team. I cannot take a drug into humans unless I've got people who understand how to measure pharmacokinetics, unless I've got people who can do the modeling for pharmacokinetics and for pharmacodynamic interactions, people who understand the regulatory space. So in contrast to the academic scene, where it's all about what used to be all about the individual investigator and their money and their funding, it was about working as a team to bring something to a goal. So for me, that was it was an excellent transition, arguably one I might have made a few years earlier. So I had 8 years with Novartis working on a wide range of inflammatory conditions, not limited to just pulmonary medicine, and then was getting a few gray hairs and needed to think about moving back to the States. I was getting to the point where the Swiss would force me to retire, because you get to a certain age in Switzerland, they will not let you work any longer. So I was looking for a job. My kids had done a lot of their growing up in the States. They'd all moved to the West Coast. So I started to look for jobs, and I ended up with the opportunity to either go to Genentech to continue to do translational research, or to settle in Seattle, where my kids were, and to work with the Bill and Melinda Gates Foundation. So at this point, I make yet another transition in my career now to philanthropy, and working with the Bill and Melinda Gates Foundation, who were looking for somebody to support the grantees who were doing exactly what I had been doing, taking medicines into disease, new medicines into diseases, or old medicines being repurposed in disease. And again, same principles of translational research, which I'll talk about later this week at the workshop. Same principles, different molecules, different pathways, different diseases, new challenges, very interesting and fascinating work. Again, working in teamwork. And opening my eyes to problems in global health that are much more familiar to you in this part of the world than to me as an Englishman or living in Boston or Seattle. So exposure to diseases like river blindness, human African trypanosomiasis, and my first visits to Africa, seeing the research that was being done there, learning from the investigators on the ground, a whole new experience. But again, taking a skill set that I had developed in a different context and now applying it in global health. And I have to say, it was a very satisfying part of my career. The foundation also, as you can imagine, educated me as a human being as well. I was fortunate to be there when Melinda Gates was one of the co-chairs. And Melinda is an extraordinary role model as somebody who was really working very hard to empower and educate women and girls, and made sure we had had that lens on the research we were doing. And it wasn't just a bunch of old white men developing medicines and deciding what other people should do with their lives. It was a very good education personally as well. So I think if I were to look back on that, I probably had 3 different careers: a physician, a scientist, and a drug developer. They've all been very rewarding. I think when you're trying to, don't be afraid to make changes. We've already talked about that today. When you do make those changes, think about where it's going to take you. It's not just the next job you're going to do. Where will that job lead you to? If you can do it, manage it. And I think it's the ideal. Like what you do and do what you like. In fact, I'd say love what you do and do what you love to do. And I think, one of the things that Frank would say to us is people tend to be good at the things that they love to do and vice versa. And so think about that when you're thinking about what you want to do with your skills. I'm going to stop there and take any questions folks may have. Oh, I'm just going to answer the question I had put earlier to Ju. I was blown away by the quality of the science at Novartis. I had seen this before as an academic when a lot of the basic science research on leukotrienes was done by Merck. And you look at early publications and the identification of leukotriene B4, leukotriene C4 and its receptor, all coming out of Merck. Superb work published in top-rate journals, Nature Science, and so forth. And I'd seen this with friends who were working at Biogen when it was a biotech company before it became a huge, larger pharmaceutical company. Superb work coming out of Biogen, papers in Cell appearing on cloning of VCAM, for example. So I think there's superb work. Some of it doesn't see the light of day, but what you do see is superb.
Thank you very much. It was very nice. I don't know if we have any comments here, but I have one point that I would like you to comment about. That the training opportunities that no, just sorry. About the training opportunities that people may get in the pharmaceutical company, comparing with the academia, and also in your point of view, how do you feel that you could impact the younger people that you were working with in the pharmaceutical company, comparing to the other work that I'm witness and I benefited from this opportunity to get my training from you?
That was a good question. I mean, to your last question, Bruno, when I was working at Novartis, I didn't have my own lab. I had the opportunity when I moved to Switzerland for 4 years to do that, but I chose to focus on leading a team of translational investigators. And at that point, the influence is a little different. At that point in my career, I was very much focused on finding good people and giving them the resources to do what they do well. And one of the things that I hate is seeing people in seniority thinking they can meddle in what's happening on the ground and with what the team is trying to do. It rarely works because it's the same as in the military. You're not trying to. People on the ground know what's happening on the ground, and you need to find a way to make that work out. So I think my influence on them was really just, it becomes a question of how do you want to do the work as much as anything else. And I think we had success by letting good people do good things and do what they're good at. I think the training opportunities, 90% of training is what you do yourself with your hands. I think the formal training in the pharmaceutical industry, at the time I was there, they actually think about leadership training, which we didn't do in academia very much. And I think I had the benefit of leadership training as well. I remember one course which was very valuable in my career was essentially how do you persuade people to do what you want them to do if you have no authority? I mean, they called it leading without authority, but that's what I did almost all my time. I'm in a team, I'm leading a team, but none of them directly report to me. The pharmacokinetics guy reports to his pharmacokinetics people. The regulatory people report to their people. So we are working as a team, and I'm leading them. And how do I influence them to make the right decisions? And how do we together make the right decisions to go in the right direction? So I think there is excellent science training, but that's you got your hands on the job. You're doing the work in industry. But I think the leadership training is far superior.
Thank you. Miriam?
Thank you very much, John, for sharing your experiences with us. And I think something that is probably going to come out in other people's reports and talks, but with your experience, I think you have a different view on it. And it's about the skills, and both in the teams that you have been leading directly and the teams that you have been interacting with. That was something that when I was doing my PhD, we had a lot of conversations on, and we tend to have this imposter syndrome spectrum where we always think that we are much worse than everybody else and we don't know enough. And there was this thing about how studying science and having the scientific method can help both if you're in lab, but also if you're related, or sometimes not even that related, but you can help manage and help solve problems. So I wonder, when you're leading and also doing with your work on the foundation, what do you think are skills that you appreciate that maybe people can relate to and say, oh, maybe that I can do. Nobody told me that would be that relevant, that could help me go up, or that I should work that a little bit better. And mostly on what you said, like we are trained to pipette. We are trained to ask questions, but sometimes there are some skills that we need to have to be able to analyze the data, but nobody tells you, oh, that's awesome, that's right. And I wonder if there's anything on that end that you could, that you think of sharing with us.
I'm tempted to ask you what skills you're thinking of, but for some of it, particularly when I was training, if I needed a skill, I went out and learned it, and I wasn't afraid to do that. Nowadays, many of the skills we need are so sophisticated. When I learned statistics, there was this little book, really little, called Statistics at Square One, which the British Medical Journal published because they were so frustrated with people designing clinical trials who had no idea about power calculations, for example, or how to analyze the data. Nowadays, if you want to do power calculations, you're going to run simulations. I don't know how to run a simulation to do that. The same with the statistics. The statistics, not just in power calculations, but the analyses. Everybody's into Bayesian analyses rather than frequentist approaches. So I think part of the answer of that is also learning to work with people who have the skill set you need. I came across that when we had a data set for gene expression. I had no idea how to analyze this in a meaningful manner that's going to be publishable. So you need so part of it is not being afraid to get out and get the skill set. And part of it is appreciating what other people do so you know how to use it, but also building the collaborations. I would say that I've been very fortunate with the skills I've developed as a physician, as a scientist, both translational and at the bench, and then developing medicines. I remember when I got a job offer to go to work with Novartis, I was in my mid-50s, and I was talking to my sister-in-law, who's the same age as me, and Jane says to me, oh, isn't that nice? You can get a job at your age. And, we have this idea that somehow, you get older and you're just making way, get rid of the dead wood, make way for the youngsters. But I have found that because I've developed a skill set, it's been very much in demand. So I think, again, don't be afraid to think about what skills people need. And where do you think as they say in ice skating, don't skate to where the puck is, skate to where it's going to be. Where do you think science is going? What skills are you going to need? If you don't understand anything about artificial intelligence and machine learning now, you need to. It's easier for you to learn it than me. But I think, there's just, and that's something we should have been doing 5-10 years ago.
Thank you. Yeah, I think I was thinking about that, especially this the thought that you can look for information. I think that's something that we do daily, right? If you don't know, look for a paper and cruise through what Google tells you and choose what you think has good basis. Be critical and independent on what your mindset is. If you're critical of your reading, right? And I think we practice every day, and sometimes it's not as trivial. Like we meet people that are outside of science, and it's not natural to them. And I feel that is something that it can work in any field. And so that basically that's kind of what I was thinking, but was exactly what you said is if you don't know, look for it. You have the tools, and if you don't, you find somebody who does, and you're nice to them, and you ask them to teach you. And if you do it well, they're going to be happy because, as I tell my students, that if you want to make a scientist speak happily about something, you don't even need to ask them about their kids. Ask them about their science. They will talk as passionately as they would if you ask if their kids are doing well, right? Thank you, John. I think now we would have Emiliano. Is it Emiliano? Are you there? Sorry, Victoria. Can you hear me?
I can hear you. Hello, Miriam. It's so great to see you.
Hello, Victoria. Thank you so much for joining us. In the beginning, we mentioned we wouldn't be introducing each one of you because the point of this discussion is for you to introduce yourselves and to talk about your experiences. So I would just ask you to share with us what you were thinking.
Sure, absolutely. And just thank you for the invitation. I do really appreciate that. And I've really enjoyed listening to the other speakers. I can relate with so much of what John said. And also, please teach me how to pronounce your name. Giuliana, is that correct?
It's Juliana.
Juliana. So, and one thing I think, one thread that I'm hearing that kind of runs through everyone's experiences is that need to, first of all, just learn how to utilize all of your resources, right? And that need to be a good collaborator. It really helps you throughout your academic career and your professional career. So I'll just backtrack and just let you know, my name is Victoria Love, and I'm an older person. I had a non-traditional educational background. I joined the Navy when I graduated from high school, and did get an honorable discharge from the U.S. Navy. And decided that I wanted to go to college. I had always been interested in science as a young person, that and also English literature. So I went to San Diego, which is where I grew up, and ended up going to a community college because I was a first generation college student. No one else in my family had graduated from college. So that was my path, was to go to a community college. And I joined a program that was sponsored by the National Institutes of Health called Bridges to the Future. And I had an opportunity to work during summer doing research, doing biomedical research taught by just an amazingly talented professor who, it was his life, Dr. Paul Paolini from San Diego State University. It was his life to just train and encourage people from underrepresented backgrounds. And there I found a community. And I learned a tremendous amount. I worked in a Drosophila laboratory at SDSU as an undergraduate. I met a friend who was also part of the program who was from La Paz, Mexico. He is from La Paz, Mexico. And he was working at the Scripps Research Institute. And he asked me if I would be interested in joining his immunology lab with him. That was led by a person who I'm still in touch with, Dr. Ann Feeney, who is a molecular immunologist. And it was perfect for me. what I had been learning in my classes about antigen presentation and T cell responses, BDJ recombination, and lymphocytes, it just kind of all came to life for me in Ann Feeney's lab. And as an undergraduate, I had an opportunity to publish my research on BDJ recombination. I knew I wanted to go to graduate school and ended up going to Harvard Medical School. A lot of people have an assumption that they must come out of their undergraduate work and go into a master's program before they can then move on to a PhD program. But that's not everyone's path. Some people choose not to do the master's degree. While at Harvard Medical School, I decided to join Dr. Andy Lichtman's lab. And Dr. Lichtman is a pathologist, still at Harvard Medical School. And his lab was really interested in understanding the immunological mechanisms behind certain human diseases such as myocarditis and, of course, cancer. And while I was in Andy's lab, I worked with a couple of different postdocs, and I did set up a transgenic system adoptive transfer model for melanoma using the OT1 TCR transgenic system. And the T cells from those mice recognize chicken ovalbumin, which you can overexpress in whatever tissue you wish if you have the right promoter driving the expression of the experimental protein. And what we chose to do for my thesis project was to understand the role of CTLA4, very famous molecule, that was overexpressed specifically in CD8 positive T cells. Arlene Sharp's lab had done a tremendous amount of work characterizing CTLA4, but a lot of those studies were done in mice that had a knockout of that protein, and not just only CD8 cells, but all CD4 cells as well. So it wasn't clear what influence the killer T cells would have in the absence of CTLA4. It was quite a very involved genetic crosses, a series of genetic crosses that I performed to produce a novel animal in which CTLA4 was only knocked out in the CD8 cells. And those animals, although they did not express CTLA4, the lymphocytes were not hyperactivated and could not be lethal in those mice as in the original strain. And the reason for that is that they lacked the ability to become activated unless they were exposed to their cognate antigen. So again, the T cells from those CTLA4 knockout mice had to be harvested from animals and then stimulated in culture with their cognate synthetic peptide. And fortunately, we did see a phenotype in those CD8 cells. I did publish a paper on it. Long story short, when you adoptively transfer activated CD8 T cells into a recipient animal that expresses ovapeptide on tumor cells or in cardiac myocytes, those CD8 positive T cells that are lacking CTLA4 cause disease in those animals or reject tumors in those animals. So they reject tumors in tumor-bearing mice, and they cause myocarditis in animals that express the protein ovalbumin in cardiac myocytes. That was a lot of work. During that time, I also had a child. I was in graduate school. Lori Glimsher was on my advisory committee, and I was able to complete enough work to defend my dissertation so that I could graduate and move on. Decided to stay in Boston for a little while. And joined a viral pathogenesis lab where I also wanted to understand more about T cell activity, but in a rhesus macaque model of SIV. I decided not to stay in Boston because I had a child and my husband and I wanted to go and be with my family in San Diego. I was fortunate enough to be hired by Pfizer in La Jolla. And I completely believe that happened because I chose to study immuno-oncology in graduate school. That was a very smart choice for me, but not something that I actually planned. And it worked out to my advantage. When I went to work for Pfizer Oncology in La Jolla, I worked on a lot of the same targets I was interested in graduate school, including CTLA4, but also some other new ones, including 4-1BB, which is another protein that's expressed on activated T cells. The team that I worked for at at Pfizer was just excellent. The person, our team leader, every sense of the word, just a very well-trained scientist. It was a little bit of an adjustment for me to learn how to conduct science in a pharmaceutical setting rather than in an academic setting. In an academic setting, you have more of a liberty to really research an idea that you might have. If you do an experiment and you have, an interesting finding, you have the liberty to kind of follow it up in an academic lab. When you're working in a pharmaceutical lab, the bottom line is to get the data package together so that you can advance the program. So that was a little bit of an adjustment. Fortunately, I was able to work on a project when I was at Pfizer that did go into phase 1 clinical trials. We developed a biotherapeutic antibody against 4-1BB, and all the preclinical data was excellent. Had a lot of efficacy in all the models that we tested, and we were able to get together a successful data package and go into clinical trials. Unfortunately, the program was not able to progress past phase 2. The antibody did not work as a standalone antibody for all the different types of cancers against which it was tested, and also did not fare well in combinations with chemotherapy or other immunotherapies. So that program was discontinued. When I left Pfizer, I went to work for a small biotech company also in San Diego that was also looking at making antibodies in vitro that they could then use different methods to have these antibodies be just highly specific for their targets, and they were able to do in vitro hyperaffinity mutation. Excuse me, that's not the correct term, but they were able to have developed very mature antibodies using their in vitro systems that were superior to using, say, hybridomas. I chose to go into medical writing rather than stay as a bench scientist in the pharmaceutical industry. And the reason for that was because of my family. When I was in grad school, I had my older child, and then I had another child as well. And I thought that medical writing was a good choice for me because I could combine my love of research and science and biomedical research with my interest in writing, which, is something that I had always enjoyed in my life. I think that it was a really great career choice for me. Other people are talking about work-life balance and flexibility. And while deadlines are real, and sometimes you just have to put your nose to the grindstone and really focus in to get a project completed, especially if you're working on dossiers to regulatory agencies, the timelines can be quite aggressive, and there's a lot of technical detail that's required in order to get a very good package together. And I can attest to working very closely with other people who have other expertise in terms of your PK scientists, your safety scientists, pharmacovigilance, biostatistics. Everybody works together as a group. It's impossible to do science in a vacuum. It's also impossible to be a medical writer in a vacuum. Right now, I did work for Thermo Fisher Scientific for several years. When I worked for Thermo Fisher, I worked mainly as a writer looking to reach customers who wanted to understand the basic science behind how the products worked. I was supporting the antibodies and immunoassays, as well as the imaging. The imaging groups. And I worked for a marketing team. So my job was to write all the content to help people understand how the various antibodies and immunoassay kits and instruments could be applied to basic research. And then I switched over to regulatory writing, which I'm still doing now. And I work for a company called Celcuity that's based in Minneapolis, in Minnesota. And interestingly, this company in-licensed a molecule from Pfizer called gedatolisib. And this molecule, it's a PI3K AKT mTOR pathway inhibitor. So it hits, this small molecule hits all three of the nodes in that pathway. And it's the preclinical data and also the phase 1, phase 2 studies have shown that it's superior to agents that hit any one single node in that pathway. Celcuity is now running phase 2 and phase 3 clinical trials in HER2 negative breast cancer, HR negative, HER2 negative breast cancer, and also in prostate cancer. So there's a lot of activity ongoing right now as we are wrapping up some trials that have been ongoing. I'm not at liberty to say exactly what the company is focused on for the future, but for me, I think it was a really great decision to transition into medical writing. I'll stop there and ask if anyone has any questions.
Thank you, Victoria. Bruno Diaz here. Miriam had to step out a little bit. We are doing everything, not doing a good job doing it. But anyway, thank you very much for your talk. It was really interesting. And I guess I have a question for you. Do you miss the laboratory? Do you miss doing bench work?
You know, for a long time, I did miss it. I truly miss that whole process of being able to conceive experiments, figure out what's required to conduct those experiments, being able to analyze the data and work with teams. For a short time, I left medical writing and went back into discovery research with a small company, Systems Oncology, that was also using machine learning to look at cancer signaling pathways. I didn't get to do bench science while I was at that company. Instead, what I did was to help my colleagues devise experiments and then resource the work by hiring contract research organizations, also working with academic institutions. Although I wasn't doing hands-on work, I still got to participate in the spirit of research. To answer your question, I probably wouldn't go back into a lab at this point in my life, but I think that there are so many ways to stay connected with science, even if you're not, doing full-on research. There are lots of ways to make contributions.
Sure. No, and that's what I find mostly interesting. That's I see people that really enjoyed the whole scientific endeavor, but they are not really into doing the bench work, and especially that part that everybody here knows, that when it does not work, right? So if we could just skip ahead to when it works.
I would like to say something. I apologize for interrupting. So part of the responsibility, though, in hiring other scientists, outsourcing the work, is you're still reliant on having those experiments be successful. And you're still very much a part of evaluating the data and also participating in the troubleshooting. And it can be honestly a little bit frustrating because sometimes people might not be able, for whatever reason, to carry out the instructions exactly the way that they need to be. And you know that if you were in the lab doing it yourself, you would be triple, quadruple checking everything. So it can be a little bit daunting, but when it works, it's rewarding.
Right, but is there any way that anyone can recognize that their skills could be better applied, or even their efforts, right? In what you are doing, that you are in the scientific efforts and participating, but you are not directly involved with performing the experiments. And that's I ask that because maybe we have some people here that actually feels like that. Feels like I enjoys a lot science, but not so much the more practical part, and how they can actually see if it's something that would fit them. Is there any sort of specific training or anything that they can try their hands if it's something that they would enjoy doing?
So do you mean a research scientist working at the bench, would they have an opportunity to maybe be part of a team that is focused on developing a product or doing research, but they're not, themselves conducting the hands-on work?
I was thinking more about your current stage of your career, about the scientific writing. That's I would think that some of us here would really enjoy doing it. But how? Because it's not easy to imagine that this kind of jump is possible, and you are the living proof that it is possible. So we can all do this. It takes. How can one tell if he will be good doing it?
I understand. So there are so many resources available, and I can give you just a clue about some of the resources that are available to anyone. And then what I can do is put together a list of resources and give it to Miriam, who can then share it with people who might be interested in pursuing or at least thinking about medical writing. To be honest with you, Stanford Online does a course called Writing in the Sciences, and it's free. I don't know how frequently they offer that course. That isn't necessarily specifically for medical writers. It's for anyone in science who wants to improve their writing skills. But I think that that's a really great course because it allows people to understand their aptitude. And even if they decide not to pursue medical writing, they can still improve upon the skills that they already have. So I would highly recommend that. And then there are LinkedIn, for example, has some courses that people can take if they just want to get general information about any particular field, including medical writing. So, and then there's the American Association of Medical Writers, AMWA, free online. Anyone can go to their website, learn about medical writing, because I work in a very specific area of medical writing now, but it's a huge umbrella. So there's something for everyone. you can even become employed as a medical writer and just only focus on writing research papers or manuscripts for publication. There's just so many things that can be done. So for anyone who just wants to kind of take a look, there are free online resources, and I would start with AMWA, but I will give Miriam some information for your students.
All right. Thank you very much. It was
Sure.
quite insightful. Thank you. We are actually, I'm not sure what we are going to do next. Miriam left me, just holding it. Not sure what happens in sequence. It's a break. Yeah, we are going to break for a while. We are going to have a coffee, but we'll be back soon. I apologize. I see that João is already in the room. We'll be back shortly, João. Thank you very much.
Bruno, you are mute.
Hello. Hello, everybody. Back. I was talking with muted microphone. Emiliano, how I would like to proceed? Are you okay proceeding in English? Shall we?
Okay.
Sure.
I share my screen.
Oh yeah, perfect. Thank you very much. I really appreciate you joining us today, and I must say that I've known Emiliano for long time. Emiliano was an undergraduate student of mine when I was in master as well. I was his direct supervisor at bench, but not he was not my student, but a brilliant student, and I'm very proud of him, what he has achieved, and he will talk about some of his career and his choices now. Emiliano, please.
So, hi everyone. Thank you, Bruno. Yeah, it's okay. Can you hear me?
Yes, we can.
Okay,
Perfect. Thank you.
Okay. So, hi everyone. My name is Emiliano Barreto. I'm a professor of the Federal University of Alagoas. First of all, I want to express my gratitude for this opportunity, and I want to thank Professor Bruno Dias, in particular, for training me through my early scientific life at the Laboratory of Inflammation in the Oswaldo Cruz Institute at Fiocruz. So I brought some points from my experience to talk about academia in Brazil, challenges and overcoming strategies. Okay, so I will discuss. I show some points that I believe are crucial to career in science, specifically some things I experienced to establish myself at UFAL. So I did my graduation in biology at UFRJ, and I finished my scientific training at the Laboratory of Inflammation from Oswaldo Cruz Institute at Fiocruz. In this laboratory, I began as PBIC student and finished my master's degree and PhD student in the same group. For sure, the environment and the people that period were very important in helping me develop my career. For this, I would like to thank my supervisor, Professor Dra. Patrícia Silva, Dr. Marco Aurélio Martins, and Dr. Renato Balão Cordeiro, as well as to Professor Bruno Dias and Cristiane Bandeira de Mello. I show here now our natural products, Catarina and Julia. Work at UFAL gave me an opportunity to have several friends. All my graduates are now my friends. In total, I had the opportunity to contribute to training of 24 master's degree and 11 and 12 PhD students and postdocs. So the professional career required that you to have make decisions. First, what to do after the postdoc? The first questions. Okay, but we now looking for new opportunity. In general, jobs after PhD are show come as academic jobs, postdoctoral position, or industrial jobs, consultancy jobs, and others. But we need some choice. First, which way should I go? This is a first question, important first question, but we need sure of what you want. Stay here, or where are you? In the same lab, for example, depend many times on factor beyond your control. So the second way will make your way. It's a very exciting way. So after choice the way to go is important. Just a second challenge. Are you sure what you want? Because in Brazil, the academic scientific life require carry out several activities simultaneously. It's very hard, and we need to maintain security funding. We need to publish in high impact journal. We need to establish a research team. It's very difficult sometimes balancing work and life with family. Sometimes we have holidays. Sometimes in Maragogi we can swim in swimming pool, not so swimming pool, but it's not frequently. So the second challenge will we go to comfort zone. The question is, are you ready to leave your comfort zone? When you leave your comfort zone, we discover an ocean of new opportunities. So understand, as ocean has been a new lab and new institution, a new lab and new institution in another state or another country, maybe. But you must leave your comfort zone. So moving to comfort zone, we can observe several steps to move toward the growth zone. In comfort zone, we make a resting in control, zone with safe, low risk, relaxing. But you need to move toward the growth zone to high self-esteem, to have a new vision, to increase the motivation, achieving new goals. So naturally, step outside your comfort zone is no easy choice, but is essential for maturity and experience. So for me, I list some points that are considered important to beginning of the academic scientific career outside your lab formation. So first step, preserve your scientific interaction with a research group where you formed. Maintain into network contact. Have you built through your academic career? Open new collaborative networks. Include your new student in your collaborative network. Try to understand the dynamic of new workplace and region, and identify the strengths, weakness, threats, and turn everything into new opportunities. Interact with senior research scientists in your new place. Identify this is very, very important. Identify the infrastructure available to start your project, the real infrastructure and available to use. In the new workplace, your first project should be in line with what you learn during academic training, master and PhD students, in terms of techniques and methodologies. As a young researcher, dedicate yourself totally to activity involving the research, the training, your human resource. This formation is very, very important because you need forming a new group, new arms to work with you. Become a member of a Brazilian scientific society to which your line in research belong. Being an active member of this society. Don't be immediate. The results of your action will come over time. More time, 4 or 5 years, I think, depending on the environment. So keep your scientific other things important. So keep your scientific literature updated, stay updated. And literature, new techniques, a new approach. Be creative and share your reviews about ways to do work. Develop your problem-solving skills. Maybe it is a more important point I'd suggest to youngers. Develop your problem skills. Opportunity is open frequently. Is crucial to looking for information in scientific society and media and journals on the Instagram or the website of the universities. Here we show several new opportunities in Northwest, north from Brazil, and the south of Brazil. That university this year launched in public tender. All universities had open in health-related professions this year, such as pharmacology, microbiology, physiology, biophysics, biochemistry, genetics, molecular biology, cell biology, morphology, and all. There are opportunities is frequently open. We need looking for. So the message is too important. You make your path to walking. In new lab in new place is important to pavimentate this way to others walk. So these are very illustrative in my image. Okay, so to finish, a message is important to excite and motivated the people. I would like to thank you for leaving a thought. Is a very, very important thinking. So, Bruno, this is my contact. I thank you again for the opportunity. My email and the Instagram at the lab and the website. Thank you.
Emiliano, thank you very much. I think were really good insights of what it takes to really have a successful career outside of the major scientific centers. And here in Brazil, they are really concentrated in the southeast, so Rio, São Paulo, Minas. And you made this decision to move to Alagoas. And you did not mention, but I was wondering if you could talk a little bit about how people in your friends and colleagues, they first reacted while you were in Rio in a major research institute, that you mentioned that you would be going to Alagoas to.
Yeah, I'm a crazy man. It's insane. Don't go. Stay in Brazil. Stay in the Fiocruz. FRJ is a good option, but I looking for new opportunities. I'm looking for a place that I learn during my formation. I work hard during 12 years in my formation, master degree, PhD. I looking for it's a very difficult decision to move for Alagoas, but the fear is low than challenge.
Right.
I have very, very happy to hear. Now we have a good lab, the unique lab of the Federal of Alagoas that we can see different points of biological system. We have analyzed of molecular approach onto experimental animals. We can look for histology, pharmacology, immunology, and other facilities to develop the science and in the area of immunopharmacology.
All right. No, I know your lab. I know it's great. You are. I should really congratulate you of all you achieved. But when you were showing those recommendations for someone thinking about jumping into the ocean, it was really telling that the first 4 were about collaborations. So how do you get to a new place, and then how do you find collaborators? Who you were looking for? It's just people that had similar backgrounds. How would you choose? Who would you contact to make your first collaborations?
So when I arrived, I every week I talk in a different postgraduate program. Every week I show my skills. I make experimental manipulate the animals. I make I know to both put cell in culture. I new test now new drugs. I visited the chemistry and the people, the friends of chemistry, the institute. Here the important point to isolate new molecules from Caatinga, from Mata Atlântica, new compounds, and I tested several of these compounds in different in vitro models. So I went I was postgraduate program to show my skills, and every week I test several points. So show my skills to scientists, local scientists, looking for integration to test. So it's important. But work it with previous network is very important to me. The support that Fiocruz gave me, that support that other scientists that Fiocruz, including you, my friend, Bruno and Cristiane Bandeira de Mello, but other senior research that give me important support to start my first assay at FAO. with a long time, with the time, I started coordinate institutional project at UFAL, and we get a new equipment, equipment of big port with flow cytometer, with others equipments, institutional equipments.
Right. So you told us about recipe for success, right? Making the move, but you actually probably have been witness of some moves that did not work. Do you have a recipe also for not being successful? What's the big mistakes that people make when they make such a move as yours that really hinder the success they can have?
Don't adapt the place, the local. It's very important that we adapted your skills to needs of that local. Adaptation is very, very important.
Right.
We can see maintaining the same here don't have a same structure of the folklores. So
right.
I don't. It's a crazy thing that I need isolated mast cells, isolate work with signaling pathway, but I need adapted my skills in pharmacology to apply in the new local. I recover my techniques of experimental animals, my tests of nociceptive tests, behavioral tests, to looking for anti-nociceptive agent, painful agent. So is adapted. It's important to adapt the local real.
Right. Yeah. I recall some conversations that I had over the years of people just complaining that they could not do exactly what they were doing elsewhere, right? In an institution that had much larger critical mass of equipment, people, reagents, everything, and they just arrived in a new place that they should actually be explorers, right? And be creative and be daring and try new connections, but they are just trying to mirror what they left behind. So what's the point of living if you're just trying to mirror? And you cannot mirror, right? Because you don't have the exact same structure, the exact same people around you to do it. And I witnessed how successful you were making these new connections. People never met before. You had no connection before, but you actually were very successful establishing a research program that was both prolific in terms of publication, but was also locally relevant, as you mentioned.
Yes,
right.
Yes.
Right, Emiliano, thank you very much for sharing with us your experience. And we are going to call Professor Bernardo now. Thank you, Emiliano.
Bye-bye.
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 Hello, Bernardo. It's very nice to see you. We are really eager to hear about your experience, and I'm sure it's a nice one.
Well, thank you so much for the invitation. It's a pleasure to be here. Thanks in particular to Miriam for the message via WhatsApp a while ago. It's particularly emotional for me to be here because I was in your situation. I mean, some of you there, the students, many years back, I recognized this room. it was a really great time as an undergraduate in the Instituto de Biologia no CCS. So I mean, I'll maybe tell a little bit about how I get where I was, which was starting where you are. And one quality of the program there in biology and many others was this exposure to laboratories. So the biggest advice I would give to you in this stage is to really spend as much time in the laboratory as you can. And back then, that was enabled also by fellowships from the CNPq, and there was some even credits given to hours in the lab. So spending hours in the lab is a unique element of the education that you are having right now. Many places, and I'm here to talk about academia abroad. Students typically don't get exposure in the lab, and oftentimes they start as a PhD students, and they're very, very, very green, and frankly don't have a good understanding what kind of laboratory life feels like, and what it is that they have in front of them. So after the biology program, in this auditorium, I did a master's in there as well, and then went for a PhD abroad, which I was also, and by the way, I was also very fortunate with the mentors that I found. So I joined a lab in my second semester in the program in the Department of Ecology, and I was really, really fortunate by the training I was giving, by the generosity of the PIs, which really meant you would spend hours in the lab, digging into whatever you want to do, as long as you were there and you're committed and you use your time well, which they meant you spent summers in the lab as well. And but it was exciting. Everybody, and it was not just me, it was a culture, and I'm sure the culture is still the same. It was the culture of the place. Everybody did that, and the PIs were sort of enabled that. So take advantage of this. So after that, I went to do a PhD at Harvard, which I was also very fortunate with the labs that I found and the colleagues, the networks of peers I had. I was very fortunate to have Miriam as a housemate for many years, and there was a huge group of people who would be very committed to science. They were spending lots of time in the lab, and they were talking to each other. They were motivated, and kind of that feeds back, right? So just the biggest advice I would give you is just not just spend time in the lab, find the right PIs and right advisors and mentors, but also find the right people who energize you in that particular direction, which is to spend time in the lab and do research. And because it really is, something that takes a lot of time, and we all know it can be very arid, right? So oftentimes you do the same thing again and again, and it's a learning process, and it takes a lot of time to learn, and there is no better way to learn than by doing it and trying again and again, which is also, by the way, a good quality of the program back then, which was there were great classes and lectures, but there was also, again, enough time for people to spend in lab, and in science, it's all nice and well to find lectures, but I think nothing really gives you more than the time you spend yourself, not just reading what you have to read, but trying the experiments you were allowed to try if you have the right environment of mentors. So after that was a few years as a PhD student, and I was very lucky there too. And then after that came postdoc, which we all oftentimes do. That was maybe the most stressful time, because I think when you're a student, it's you're given, it's a privilege, right? You're given this free time, say 4 years as an undergraduate, and all you have to do is to study, and that's great. And then for the PhD, it's kind of the same. It's like a bucket of time. It's well defined. You have a bunch of peers in the same cohort as you are. So just find the right ones, make friends with them, and you're gonna do well and spend time. Spend that time well. When it comes postdoc, it's a bit more of a lonely experience, and I think it depends a lot on the advisors you have. So that transition can be challenging. And I mean, one thing I would not recommend also. I mean, again, I'm talking here about my experience, and maybe I will stop early so we can have questions. It's no, don't be afraid of transitions, and go for changes. And there are very few times that I made a change that I really regretted. However, don't make them sort of last minute and without thinking ahead, right? So for the most you can, plan ahead, but plan to change, and plan to change. And by that I mean not in 6 months or in 8 months, 2-3 years. So be always planning, where you're going to be in 3 years, and plan ahead so you can be there, not just scientifically, but also in your progression. So if you want to be in a certain lab, or you want to develop a certain skill, right? So there is nothing more frustrating than finding people who want to develop a skill in a short period of time, and which is great, and motivation is great. But depending on the skill, it does take time to build and mature and get better at it. And so just, have try and think ahead and get people around you to give you advice, so that you can be ahead of yourself and plan. So postdoc was a bit stressful, because again, it's a more lonely time. You're in transition, and and the opportunities there are sort of too many. Maybe you can go, you can go to industry, you can go to academia, you can go to country, X, Y, or Z, or you can do also different research, very different. So that was a bit stressful moment. I was lucky there too, in that I found a mentor that was interested in getting some of the stuff we were doing and writing a grant. And for me, it was an opportunity to learn to write grants, which was a very important skill when it comes to academia in the US, and I think in Brazil too. You have to, my hope was always to give people in my lab what was given to me, which is a functional lab with enough resources. And for that, you have to, oftentimes be chasing money. So I think there was a conversation here about adaptability and be ready to change. That happens also with the landscape of funding in the US in particular. It can become really, you're chasing money often, which is fine. We do what we need to do to do what we want to do, which is to be in lab and do science and have, enjoy doing that. The challenge sometimes, and the benefit, I think, the system in Brazil is that you have a little bit more stability here, because grants are so short-sighted, sometimes 4 years, 5 years, success is very small, very, 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 very limited. It can become like a rat race, and that, it puts your perspective on what you want to do and what you want to be, sometimes a bit, you cannot look as far as you would like to. When you're a student, you're given 4 years. You're in the lab 5 years 4 to 6 years as a student, as a PhD student, a doctoral student. You can plan ahead, and as a faculty, depending where you are, you can plan ahead too. Depending where you're in academia in the US, it really is a short sort of always like chasing, and that can be both tiresome and I think unproductive in some ways. The tiresome we can deal with. The unproductivity can be annoying when you feel like you're just running circles trying to get money and chasing the next sort of red ball in a particular field. And I think if you experience science in some ways, how much is loss of time and resources in chasing a particular question, right? So oftentimes the questions are not well defined. The best questions are not well defined. They're there for you to define, and the data is fuzzy or unavailable, and you have to sort of build over time. So that takes resources and waste in a way, but it's a good waste, because again, as long as you're busy and you're looking at the same thing again and again. So for my PhD, talking about changes, I was before doing in a genetics lab, sort of human primate genetics, and then I went to the fruit flies, which I had never been exposed before. I was exposed in your department there by a professor that I shared the name with, Bernardo, and that was great, but I never had actually done research with it and played with those flies. But spending hours doing it was great. You just have to, it's a good waste. It's a waste of resources and waste of your time, but it's not a waste, right? Because you're really using that time to learn and to learn the possibilities of that model and what you can do. And there's no better way to learn. Again, reading books is one thing, but when you're really trying things on your own, that's when learning happens, and you can come up with new ideas and new projects, and being in an environment that enables that is wonderful. So after that, I started as a faculty in a very different environment. I was a PhD in evolutionary biology, and I was a faculty in the Department of Environmental Health, and that was a huge change, very, very big. And my colleagues were all worried about toxicants. So they're worried about things like PFAS or bisphenol A or lead, chromium. It's a school of public health, and you really hear all these talks about toxicants and the many things they can do to you. And it was there was a great transition for me. It was a supportive environment, and I learned a great deal about things that I never thought I would study. Right, so I was studying fruit flies, basic genetics before, and now I was studying toxicology and environmental health. So it's been great. And after that, I started studying aging, and now I'm at the University of Arizona and directing a center on longevity and neurodegeneration with a focus on Alzheimer's. And we start studying epigenetics and aging clocks. So maybe I'll stop here or slow down here and take a pause and see if there are any questions or directions that you'd like me to go.
Hi, Bernardo. So thank you again for accepting and for giving this overview. And it's not really directing, but I think one topic that comes about a lot is how to choose. You mentioned about moving a lot, and that is nice if you are thoughtful. And what do you think are the criteria that you thought were more important? Because let's say when I was moving, I was looking at the topic of research. But sometimes, as I said, you get there and it's not a supportive environment. And so what type of questions do you tend to ask? Or what questions did you think were the most helpful? The answers were most helpful for you to find the right environment for you. That might be a nice point of start for somebody who's trying to make a move now and thinking of the next place to go.
Yeah, so that's a really great question, Miriam. How to choose? I think ideally, I would say how to choose and how to move. I mean, choose carefully and move slowly, I would say. So just sometimes something shows up in front of you that is great, and you can make a decision right away, right? So and that obviously the right decision. But more often than not, you're in a situation that, like I said, you plan ahead and make a plan for 3 years ahead of you. And be careful in those choices. And as you're making those choices towards that 3-year plan, you can make non-committal choices, if you will. You can make choices in directions that don't tie you up or don't close doors 100%. So I would go with sort of that mantra. And I mean, I was unfortunate that I never had a situation where it was really like desperately terrible that I had to run away from. So sometimes you are in a situation where there are these push factors, right? So you just really want to get out of there as soon as possible. So as soon as you recognize that, I think, and I've seen friends and colleagues in that situation, then you have to get out of it, I would say, as quick as you can. But that's like, it's obviously there is a push factor. It's terrible. It's not going to work for you. It's not working right now. It's not going to work in 3 years. So don't be stubborn. I mean, be stubborn with science, but in science, you need some stubbornness, but sometimes we get too stubborn with our colleagues and friends and advisors, and sometimes it's just not working, right? And you rather say, look, it's not working this direction. I'm gonna go in another direction. Hopefully the people around you will be supportive of that change. I think most often than not, you'll be surprised that people are. I mean, of course, some are not and a bit strange, and if whatever feeling they're feeling, say jealously, or they don't want to let you go. But it's rare that people are really sort of vicious and unpleasant, and you just get away. If the situation is not working, get away. Now, where you go, if you're going to run away, just run the risk of running away too fast in a direction that's going to be bad as well. And then it's just like bad over bad, and it builds over time, and it becomes a negative cycle. But you can make a choice, at least temporarily go somewhere and then think about it carefully and make a long-term plan. So, yeah, I would say choose slowly, choose carefully, choose slowly, and hopefully you find yourself in as few situations as possible when it's really bad. I mean, I found myself in some, someone also described to me the writing of grants in the US. And sometimes you have to write to collaborators, and it's the most wonderful thing, right? So I never turned down a collaboration, ever. Collaborations are really an amazing thing. It enables you to fly much, much higher, do things that you wouldn't do otherwise, and share the burden also. So collaborating is great. However, sometimes it feels like getting married, right? So if it's with the wrong person, you need a divorce, and because it's just not going to work. And I remember when I was starting as a faculty, this associate dean gave us a talk, and she recommended that if you're going to write a grant for someone else, you have to be able to leave your kids with this person and really trust. And ideally, that situation, I mean, that's a really high bar, but that's the situation you want to be in. So, people you can trust and you can work long term with, and you can deal, you can kind of not just be a transactional collaborator, but someone who will be around when things are not going so well. So I think choosing that way and choosing the people around you that have those qualities is really important.
Sir, we are transmitting it to the YouTube, and there is a question on the chat, and the question is from Isabella, and she's asking, how was for you the most challenging step when moving to the US? And did you feel there was a language barrier, and how did you make it easier to communicate and translate your feelings? And I edited the question, okay? But that's the sense.
Yeah, I mean, there was always, I think, a language barrier and a but not just language, but communication, right? So I think in science, it's always hard to communicate, even with between people who oftentimes speak the same language initially. So just keep that in mind, and if you understand that the language itself, English, it's not necessarily the hardest aspect of it. If you're interacting with people who have their best foot forward and they're trying to communicate with you and you're trying to understand each other, eventually you will, because the challenges are not just the English language. There are other challenges that make it harder. And so if you can cross that mindset and find the right people who are open and can dig with you into some problem, some question, the language barrier become less of a problem. there was some challenges, right? So when we were starting grad school, thankfully we were given 6 months without teaching, but then 6 months are really, they go by really fast. The next thing you know, you're TAing a class with in a language that you're not so comfortable with. But again, I think I took that to be a challenge just as it was for me as it was for the students. The poor students had probably had to deal with me as well. So we figure it out together, and oftentimes it goes okay. It does take some effort initially, of course. The hardest time. What was the other part of the question? The hardest.
So, how was the most challenging step of moving to the US? I think she mentioned that you left a lot of things behind, and you have to choose to go. And how maybe I edited that as well. So,
I think. I wanted to move, frankly. So it was. I like move. So to me, the move itself wasn't hard. I think the question is right. Language barrier, finding, making new friends, finding yourself in a new environment. All this is a challenge, and it takes effort. But it's also so exciting, right? It's like science. You're going to a new place. You're finding something new. You're making new friends. If you I'll try and keep a growth mindset. So the change itself to the US wasn't particularly traumatic. I think the biggest challenge, the hardest time, I think, was the postdoc, because the postdoc was a very uncertain time, and I knew I wanted to stay in academia, either in Brazil or in the US, and I also knew that as a it's hard to stay in academia, unfortunately, because there's a mentality in academia that that doesn't promote team science sometimes, and you're supposed to sort of break away from your advisor. So we're interviewing someone last week. We're interviewing someone for a faculty position here, and people have raised concerns that she is too close to her advisor, which is a weird thing to do, because you want people to be independent, but you also want people who can collaborate with one another and build teams. And I think over time here in the West, there's a trend towards professionalization of the scientific enterprise. More and more, you see people who now, they don't necessarily want to become a faculty, because that comes also with a lot of some other burdens. They want to join a lab and be a research scientist and make that a career. And I think, I certainly try to do that now in my lab. There was a time I would see people in the lab as postdocs. You stay there for 2, 3 years, 4, and then you go. And there was some push, in both directions. People wanted to go. People, they don't want to continue moving their career. I think over time, there is, for better or worse, I think for better, there is a tendency to get in positions that are more stable in labs and being part of your lab as a team. The same way you can go back to Brazil and do that. You can do that in the US too, both in industry, but also in academia. It's a little bit sometimes challenging in academia, because you're always sort of depending, to be in that position, you're depending on grants. But you can, eventually find teams that are well-funded, and people with the mindset, they will start teaching you how to write grants yourself. And then you can be part of that team and think about these grants as a shared a value that is brought to the team. So I think, and I learned that in the prior place I was in public health, there were lots of human cohort studies. There are studies that are done over many, many years, multiple grants, decades, in teams that they have to be somewhat stable. And there is more pressure there for people to be team workers and work together. And that's great, because the laboratory science, sometimes I feel we do ourselves a disservice by this sort of, everybody has to be independent and start their own lab and somehow even fight with their prior advisor to establish independence. There is an emphasis on that, and I think it's great in many ways, because you need independence of thinking and intellectual freedom to do what you want, and intellectual space to try new things. But it's not true that just being on your own will give you that, right? You can be on your own and be really on your own and lonely. So it's a balance, and I think in the postdoc, the stage I was in at that time, I really felt like I had to separate, and that was a bit of a challenge. And sometimes over time, now the challenge becomes mine. Sometimes I try to tell people in my lab, you can stay here on a long term, and we can write and work together on grants, and we can be, in a way that is supportive to everybody and to the institution and to the people around you.
Right. I actually have a question. At what point you knew that this was what you wanted to do long term? And at any point, these certainty wavered. That's maybe I'm not, don't want to be in academia any longer. I want to do something else. So,
I knew that most strongly when I was at the stage of the people in the audience there. When I was an undergraduate, I was, really in love with science and research, and I felt like, I found a welcoming environment, and I think you find, and in Brazil is a great place for that, because of the way the funding is. You can have a fellowship from the government that enables you to spend time in the lab, and you should then use that well. And it's not necessarily tied to a grant. And of course, some laboratories have more resources than others, but you still have your sort of little fellowship, and that means something. At least you have some financial compensation. But more important, you have sort of the, it just makes you feel good, and you spend time. And if you have that ability, of course, sometimes you have to fund yourself in other ways. But spending time in lab when I was starting was when I had the most conviction, and then over time, it kind of sometimes it gets weakened. Not necessarily during the PhD. It was a good time. Maybe when it became harder, when I was starting my laboratory, and I went to the medical area in Boston at Harvard, where Miriam did her PhD, and the mindset there is can be a bit sometimes different than what I had experienced before. Not because of the competition necessarily, but it's a more corporate mindset sometimes. And not only you're in a corporate, I was coming from a very academic mindset in biology in Brazil, and then in a department that sort of prized innovation and all that. Then I was in a place that I was new there, even though it was the same institution, but not really. I mean, so different. And I didn't know anyone, and I was starting the lab, and it was supportive, but there was a lot of pressure to get grants, and getting a first grant and getting initial funding it was so hard. And I see this now, right? So the other day I saw someone, and she has a grant at the 13th percentile. So meaning she's like almost top 10, and it didn't get funded, and she's never got a grant before. It's like a new investigator. So that's depending where you are. That's where you are in terms of research direction. Funding can be very, very difficult, and even more so initially. So that was a very stressful time, and you kind of lose a little bit of hope in the system, if you will. But somehow it works out, and I see it works out for many people. And because again, you find other ways to kind of, okay, I can, my student, I'm worried because my student's going to run out of funding, but then, there's this little pilot project that shows up, and I can write for it, and, start, you have to be very creative in piecing together funding, and that's maybe the most challenging. That was early on in the grant writing process. Over time, it became more of a business. So sometimes oftentimes I miss that experience, the initial experience of having that research excitement, because now I sometimes make mistakes in the sense of being too pragmatic. I want to get grants because I want to be able to fund people in the lab, and not necessarily going for the most exciting, or what I think is exciting research directions, which, like most people, I'm wrong half the time, but it's fun to be wrong, and you learn, and you try it again, and you do it differently. But there comes a time when I can't take that risk because I'm responsible for people. So it just, I just do what I need to do to get them funded.
Great.
I think I have one more question. You mentioned a couple of times in the beginning that you're lucky that you saw this opportunity, or you're lucky that you were in a place. And I think there was something that we were talking about in the lab a while back. I think you can let's say, be more open to luck if you expose yourself, and if you read, and if you study, and if you are available, right? As you mentioned, was usual, and we did that here in Brazil as well, right, Bernardo? Both in UFRJ and in INCA, we would cross each other on the hallway any time of day, or sometimes at night, and weekend when it was necessary. On vacation, we were happy because we could stay in the lab. We didn't need to go to UFRJ to catch classes. Mm-hmm. And I think that's something that sometimes we don't see as much. People don't spend as much time in the lab. And as you mentioned, in your lab initially, the PI saw it in a happy way, and would end up giving opportunities both with projects and with going to meetings, and because you're there, and because they see that you are present both physically and in the mindset, right? And you're enjoying it. And I wonder, now that you're on the other end, right? So now you're hiring and you are evaluating people. And if you think anything different, or if you see anything different that you could say, maybe to the youngest, don't worry, right? And be there. And because at least that's what I say. And I'm not sure how you see it, because in a different environment, how would that feel for you?
Yeah, I mean, absolutely. And like I said, for instance, when you go to a PhD, right, there is no better time than that. And I tell this to students. So you really, I mean, there are no better time, because imagine oftentimes when in your life you're given 5 years of reasonably secure funding, especially as a student, not as a postdoc necessarily, but as a student for sure, reasonably secures funding as a doctoral student in Brazil or PhD student in the US or anywhere. When are you giving that opportunity again of 5 years or reasonably secure funding where you can really think as deeply as you can and as wide as you can about any topic? So just, be there and be in the lab and be with your peers and serve others, be a resource to others and they will be to yourself. So finding that is like, and that's maybe what you meant by being open to luck, you just be immersive, right? Especially, a lot of what we do in the laboratory is so immersive and so so time consuming, and it takes your life, right? So like someone, like a friend says, you kind of, the more you kind of, you go to sleep thinking about it and you wake up thinking about it, the better. But you cannot do that alone. And I think I was, in a sense, very lucky to have found, people like you, Miriam, but also others in the, in the programs that we were in, that they were just excited to be there and were using their time well, because you really need peers and it's really hard. I learned that also when I went to start my faculty position. What I was trying to do, like still my fruit fly research and maybe mix my fruit fly with toxicology and be creative about that. It became really hard because the training of everything that had to do with the fruit flies that I was studying before had to be through me. And there was no really, there were no sets of classes. The students were coming with a different mindset. And I got some people excited and we published papers and, got a grant on it. A name at least of a grant on that stuff. But it took so much effort and the transition was so hard because there wasn't really an environment of students or people who were thinking the same way. Over time, you just learn to, like someone said, to adapt and you adjust and you see like everybody thinks this, in this particular direction, they care about this and that. There's you can be passionate about a topic and there's so many times you can, go in that direction, try and push it. But there's also some value in understanding there isn't that much. There is a bunch of other people who are worried about something else. They don't care so much about what that particular thing, what you do. And that's okay too, because their concerns and their research is valid and exciting if you just take the time to immerse yourself and learn about that. But maybe it took me like 2 years before I was sort of stubbornly, I was stubbornly trying to like push whatever I was doing before instead of just break away, which I didn't want to break away. So I would never recommend breaking away either. I just want to keep, keep most things going as long as you can. But yeah, spend time in lab and trust that things are going to go all right. If you have a supportive environment of mentors, PIs, other faculty, peers, students, you find it not only be great for you, but it'll be great for them too. I think you had the same experience, Miriam, but others, and I'm sure others as well. You kind of go as a group, and I had friends who ended up in all sorts of places, in New Zealand, in Texas, and in France, and others here others in Boston, Arizona, and you just go with the flow. And go together.
Thank you. Any more questions? Okay, thank you very, very much, Bernardo. I think it was really interesting just to hear what you had to say and brought some different topics from what we had been talking about. Thank you.
Thank you all. Good to see the place and everybody.
Thank you.
Thank you. Bye.
And so, João, I think. João, are you there? Oh, hello. Hi. João, so we'll keep on this. Since you're going to be talking about your path, we are not going to make an introduction, formal introduction. So just let you talk first, and then people will see why we invited you.
Sounds good. I'm going to share my screen. Can I have a few slides?
Sure.
Let me see if we can do that. I hope you can see it. Okay, good. All right. I don't think I can see all of you. So if you need to stop me, just interrupt me at any time. So thanks so much, Miriam. It's really great to see you and Bruno and many familiar faces. I don't think I have seen Bernardo face to face in many years. Last time was in a conference somewhere in New England that I don't even remember where it was. So it's nice to see everybody. You're looking good, Bernardo. I don't know what kind of research you're doing on aging, but it's working. So I'm João Monteiro. I, as many of you, was also at the Federal do Rio de Janeiro, did my both my medical degree and my PhD there at the Institute of Biophysica. I think the last time I was in this auditorium, I was defending my PhD thesis, and this was a long time ago. It was a very happy moment in my life. So it's nice to be back in a different capacity. I have switched gears a few times in my career, and for the past 13 years, I have been a professional editor. I'll talk a little bit about what my trajectory has been since I left the bench side of academia. I don't like to say that I left academia because I think my job is essentially immersed in academia. I like my entire 24 hours of the day are thinking about science and talking to scientists. So in some ways, I'm actually more into academia now than when I was at the bench. But for the past 6 years, I've been the chief editor at Nature Medicine, which is a medical journal, part of the Nature family of journals that publishes research across the entire landscape of medicine. So I thought, it would be interesting to share a little bit of what my trajectory has been. I think every person has a different trajectory. But to me, I think the most important thing when I talk to young scientists and people who are thinking about next steps is really to look back at my own trajectory and think that careers are not necessarily, or at least they don't have to be linear paths, right? I think when you're young and you're sort of thinking about your PhD and, where you're going next, you tend to think about things on a very linear fashion. While in most cases, you make detours and go back to where you were before and move into a different direction. And I think for me, I think both my education and my professional life involved a number of convoluted steps that sort of brought me to where I am now. So my scientific career is started at INCA, Instituto Nacional do Câncer, in a very happy time there. I think it was an absolutely enriching environment. That's where I met both Miriam and Bruno. I think I was very lucky to be in that environment and to have that as a primer to become a scientist. I learned a lot from many different people. I was exposed to a huge variety of flavors of science. I think something that has been said here, there was a lot of happiness about being there and being part of that group of young people who were very enthusiastic about research and were really on top of what was happening in their fields and very willing to share what they knew and what they wanted to do next with their career. So I think that set me up to be, the professional I am today. And I'm absolutely grateful for that time there. I spent, I don't know, maybe almost 10 years at INCA between, pre-grad, medical school, and then finishing my PhD before I left Brazil. It was also because of INCA that I decided to go to study medicine. And I was very much influenced by the physician scientists that I met throughout my training there, particularly my PhD advisor, Adriano Bonomo, who was a very influential person, not only for me, but for people who surrounded her at that time. And she, after many conversations with her and deciding what I wanted to do with my my education, and the fact that I couldn't decide, I think she made a very good case that if I couldn't decide, I should go and do medicine because the opportunities and the different flavors of physicians you can be are, there are so many options that, it's a great place to be if you still don't know exactly what you want to do afterwards. And I think for me, it was kind of like that. I went to medicine because I was very interested in science. During my studies, I got in love with medicine, so I sort of developed a parallel interest for being a clinician and a physician. And for quite a while, I sort of took the two parallel paths. I joined a joint MD PhD program that I don't know if it still exists at UFRJ. I think it still does. Okay, cool. I was one of the first ones, but it allowed me to do some of my PhD while I was finishing my medical degree. It was a very crazy, I think, time of my life. I don't think there was a lot of thought about how you would organize your time to be able to do a PhD at the same time you're finishing your medical education, and that sort of it was a very intense time of my life, of running from the lab to the hospital to the lab, and in lunch times, and but was also a sort of a very exciting time of sort of trying to understand how my scientific interests were intertwined with my medical my interest as a physician. So I finished my medical degree, and then just after that defended my PhD thesis. And at that point of my life, I was kind of disconnected for the most part of the bench and from science. I went to residency in psychiatry, which was an area that was completely unrelated to my PhD, which was in immunology. And I think that decision had to do with the fact, first, I had a very strong interest in that field, but also to the fact that I had applied at that time to many fellowships abroad to go for a postdoc, and I was not successful with the majority of them. And that was a very frustrating period because, I think for what you could achieve in terms of science in Brazil, I was very well placed, but then faced with the reality of competing with people who were, for instance, in the US or in Europe and had been given access to resources and opportunities that if you are in a low and middle income country, you might not get the same level of exposure and opportunity. It really was a little bit of a setback. And then I went on to explore my medical interests. And after I was already, halfway through residency, I was awarded a fellowship from the Pew Charitable Trusts to go to the US and do the postdoc I wanted to do. At that point, I kind of had to make a decision between just becoming a physician and continuing to train and stay in Brazil, or stop that, even if it was temporarily, to go abroad and give it a try and see what doing science in highly competitive, high-stakes environment would look like. I took that path, and I left Brazil now almost 16 years ago, I believe, to do a postdoc at the NIH with Ron Germain. Spent 4.5 years there. And towards the end of my postdoc, I had already published whatever I had to publish there, and sort of contemplating what I would do next. And the options were to try to go for an independent PI position in the US, to go back to Brazil, either as a scientist or as a physician. I think those are the options that we're actively thinking about. Completely by serendipity, I ran into an editor of a professional journal. That was an editor, was a person who had actually edited my main postdoc paper. I'll talk a little bit about him later. Who introduced me to what editorial looked like. It sounded super interesting to me. And that was sort of the path for me to go into my first editorial position. And it's been almost 13 years, and it's been a really wonderful time of my life. I think it was great career move for me. It allows me to be fairly connected with science at a very high level, but also allows me to cultivate a very broad range of interests and to participate in the scientific endeavor from a different perspective. Since 2013, I've been chief editor at Nature Medicine. I'll tell you a little bit more of how that transition happened as well, but it was kind of a sort of a obvious next move in the career development of an editor. Since I'm talking about careers in editorial and publishing more broadly, I thought of sharing a little bit of information because I don't think there is a lot of awareness about what being an editor is. And I think the first thing you should know is that there are two different types of journals, scientific journals. There are those that are editorially independent, and they usually belong to either a for-profit or a not-for-profit organization, including the journal I work for, Nature, which is part of a for-profit company called Springer Nature. But you have journals like Science and the PLOS journals who are also editorially independent, but belong to not-for-profit organizations like the AAAS. These journals are generally managed and edited by professional full-time editors, which means that the people who work there, that's their only job. They work full-time, work with authors, editing papers, making decisions on manuscripts. That's their entire life. As opposed to that, you have scientific academic journals. For instance, I'm giving as an example Journal of Immunology that many of you might be familiar with. These journals usually belong to a scientific organization. For instance, in the case of JI, it belongs to the American Association of Immunologists. They are edited by academic editors who are, for the most part, active academics. They have a main job, which is to be a PI in whatever institution. And as a side job, they take a little bit of their time and then they contribute to their journal. I think that those are the two models of how journals operate. The other important thing to know is that there is editorial and there is publishing. The editorial are the people like me who are responsible for the content of what comes out in the journal, the science part of the journal, the policies, the editorial policies that affect the quality of the science of that journal. And there's the publishing people who are the people who make business decisions for that company or for that organization that publishes the journal. It's kind of like a newspaper. You know, in a newspaper, you have the newsroom who are journalists, and you have the people who run the journal from a business perspective. And for the most part, there is a wall between what happens in editorial and what happens in publishing. And the role of this wall is to preserve editorial independence in the sense that what I decide to publish in Nature Medicine should not be affected by business decisions or for whatever contingencies that the people who run the journal, who run the company are interested in what their priorities are. So the two things tend to go very separately. To give you a sense, for instance, in my work environment at Nature, we are now a little more than this, about 300 editors who are all people who had at least a PhD training. Many of them will come with their postdocs and sometimes even with some experience as PIs in academia who want to change careers. This main contingent of editors is supported by a team of about 120 people who do things like illustrations and copy editing and operations, which are sort of making the logistics of journals to work, and press officers and a number of other professionals that are supportive of making a journal happen on a weekly basis or on a monthly basis. In terms of what we do as professional editors, we tend to think about our jobs in three spheres. So we think that what we do is a little bit of curation. We look at the entire landscape of what's being produced in terms of scientific knowledge, and we then go and select what we think within the context of our own journals might be of higher interest to the readers of that field and that journal. We think a lot about who reads us. As a scientist, you think a lot about scientific publishing from your perspective as an author, but when you're an editor, you have the different stakeholders. You have the authors, you have the reviewers, but the most important really are the readers, which are a much larger number of people, and you tend to make decisions about what your journal publishes based on what kind of readership it is intended to serve. Then you do that curation, then you enhance the content through peer review process, improve the work technically, improve it the way it's presented in many different ways, and then there is the amplification part of the job, which is presenting that piece of work to the world through your journal, but through promotion in social media, through press releases. So I think that sort of covers the entire, let's say, life history of what editors do. And we think a lot about our journals having a role in cross-pollinating ideas and bringing people with different expertises and different interests together in providing visibility for things we think are interesting and could be transformative to fields. We use our journals as a platform to provide thought leadership on topics and problems that we consider important and to surface important advances. On a very practical, I would say, not very granular way, the way we divide up our time for, I would say, a regular editor, you spend about 70% of your time reading papers, discussing them with your team of editors, authors, reviewers, and commissioning content. You can think about reviews, review articles, perspectives, opinion content of different flavors. You can think about it as a very endless journal club that begins very early in the day and goes throughout the entire day and never ends and starts again the next day with a completely different set of topics and papers. 20% of your time is devoted to being out there in the community, going to conferences, visiting labs, research institutes, talking to people about what their research interests are, identifying new reviewers for your journal, meeting early career researchers, trying to understand what the state of the art of a field is and what questions people are interested in at a given time. And we spend about 10% of the time working on other projects, such as policy development, editorial policy development on, for instance, to improve reproducibility of data, to improve reporting standards, organizing events, and doing like a number of miscellaneous activities. We spend a lot of time on projects involved in promotion of open science, and that includes sort of development of tools to facilitate sharing of data, sharing of code, sharing of materials, protocols through our journals and through accessory elements that our journals tend to offer. And we also spend quite a bit of time thinking about research integrity and how we can make sure that the research we publish is sound and developing better standards that help to hold people account in the way that they do research and the way that they report the research that they do. And, some of our journals, mine include, also spend quite a bit of time identifying topics that they consider of high importance to readers and commissioning content, which might be of journalistic flavor or can be of academic flavor, so the forms of reviews and perspectives, and working with authors on some of these pieces. I'll give you here a couple examples of topics that we recently covered in my own journal. Should it just keep going, or do you want me to take a moment for questions?
Hi, João. I think that we had a peek, and I think you're going to give some tips, right?
Okay.
So maybe, I guess what we would ask would be tips. So maybe we can wait for you and ask later.
Sounds good. Yeah, I was watching some of the previous talks, and I think a lot of the tips I was going to share are shared by people working on different aspects of the scientific enterprise. I think the first tip that I have for people based on my different, my very non-linear career path is choose wisely who you will work with. I already said that I was very fortunate to have started my career in a place that was a very embracing, very vibrant place. And throughout the sort of the different stages of my career, I was also driven to work with people, not that just I admired, but people who I felt could offer me something beyond the science, right? I mean, all these people that are depicted here are people who I learned many, science hard skills from them, actual knowledge in the fields I was interested in. But they also taught me other things that were very critical to take me to the point where I am. For instance, my PhD advisor, I think, taught me a lot of immunology and science, but I think more importantly, it taught me how to think critically about any topic that I would be interested in. And that to me is sort of the most important skill that you have to have as an editor. You will be dealing with science nearly across any field of research, depending on the journal you work for, and you need to be able to critically evaluate that and communicate with scientists and about the science, regardless of the topic. My postdoc advisor at NIH, who also taught me a lot of science, also taught me how to be successful in the competitive environment, which is not necessarily something that we think a lot, or that at least was not something I thought a lot while I was in Brazil. When I left Brazil, I went to a very big lab, was a very high-profile lab and very, very competitive. Not so much, not just in terms of the competition for resources, because we had a lot of resources, but I think the competition of ideas and the competition of making sure that your intellectual how can I put that diplomatically, your intellectual priority is respected, and that people recognize what your contributions are when you're working in a larger group. So I think that to me is one of the most important skills that I learned from my time at the NIH, is that how you sort of defend yourself from intellectual, how you place yourself and your value from an intellectual perspective when you're in a very competitive environment. When I moved to my first editorial job, I think I also learned a lot of science. I worked for Cell, it was my first journal. At Cell, the philosophy was that any editor would need to be able to handle research in any field of research that the journal published. So in my almost 5 years at Cell, I handled papers on everything biology that you can imagine, from very basic molecular biology, mechanisms of transcription, epigenetics, to immunology, which was my field of interest, to neurocircuitry of flies, to metabolic diseases and cancer, to plant hormones and you name it. And I think sort of being able to navigate research and how you are able to appraise research regardless of the field was sort of a very important part of my training as a Cell editor. But beyond that, I think one of the most important things that I learned there was how to communicate effectively with different stakeholders, and not just effectively, but empathetically and diplomatically, which is also a skill that's very precious to editors. And later on, when I moved to Nature Medicine, which I'll tell you also a little bit how that happened, I think the person who I worked most closely with, who was a former editor at Neuron, which is a neuroscience editor journal at Cell Press, and then had moved on to a higher position at Nature. And her name is Meredith Lemesurier, and I think she was the person who taught me how to work with people like in a complex team environment where I had to lead, and how can you motivate people to deliver the best that they can, and to work together and to synergize with each other to produce something that's of very high value. And I'm mentioning all these things because when you're going through your training, you tend to think a lot about developing your hard skills. But I think hard skills can only take you so far in your career. When you enter the job market for whatever career flavor you decide to take, you're going to be competing with a lot of people who probably have the same hard skills that you have. So I think the soft skills that you bring of yourself make a lot of difference in that final decision that a hiring manager will make in choosing between you and someone else. So I think at least being aware that soft skills are important and trying to get some of these soft skills from the different people you're interacting with. People are better at different skills, both hard and soft. It is something that, for instance, I would like to have known before I started my scientific journey. And I encourage everybody to think more actively throughout their training period. And by soft skills, I mean things that are transferable regardless of where you go, like communication skills, how you manage your stress. I mean, a lot has been said today about, all of us have stressful careers and go through stressful moments of change, and how you manage that without burning out, for instance. I think that's a very important skill to have. How you manage your time. Are you prepared to do public speaking? I think the scientist of today that's successful needs to speak in so many different platforms and be able to communicate to a range of different stakeholders, some of them who are scientifically trained, but many of them who are not, and sort of being able to navigate this sort of complex network of people with different levels of scientific knowledge is really important. How you develop your own leadership skills, which will be applied to managing your lab or working on a team environment in industry or whatever other path you decide to take. How you deal with conflict in the work environment, and how you navigate conflict in a way that it's geared towards resolution and not amplification of the problem. And how can you optimize your decision-making skills? So I really encourage people to think about what soft skills they have, what the ones that they are stronger at, and which ones they had to develop further, and then take really a more active approach to developing them throughout your career development. I think together with that, and also it was a topic that came up in some of the previous speakers, I think in the current environment we live in, you need to be able and be open to embrace change and to adapt to whatever situations you are thrown in. Careers are much more dynamic today than they were 30 years ago. Maybe a generation ago, you could just go and take a job in academia, and that will be your job for the rest of your life. And you can choose that line of work, and that will be it. And I just don't think that this is the reality in science anymore. You need to be able to change and to embrace and to be open to opportunities that you might not have predicted, but that came your way, and you should make the best out of them. So sort of this, I'd say, embracing change attitude is very important to be successful, regardless of where in the scientific enterprise you are. And I'd like to sort of give an example, for instance, of how I became an editor. I did not think about editorial as a possible career. And completely by serendipity, I met, as I said earlier today, the person who edited my paper at Immunity, and in a conference, and we ran into each other. We had spoken on the phone a couple of times, but we had not seen each other in person. He was very fond of Brazil and Brazilians. So I use that as an entry point, and I just started asking questions about what he did, and learned more about that career path. And found it super interesting to me to be able to, go to a topic to the other and sort of have this wonderful, gigantic network of people that you interact with, and being able to see all the research as it's produced, and have a sort of an active role in helping people to decide what is important and what is not important. And from this first encounter until the moment I got my first job offer as an editor was really a matter of a few months, and that completely changed my life and my career in a completely different direction, and probably would not have taken that path if I was not open to perhaps follow a different route than the route that I had planned for myself from the beginning. So I think being ready and being open to opportunities is a major asset you can have. I also think it's very important to actively build a network of contacts, and the broader and the wider that network is, the better it is for you. And as much as building is important, I think also not burning bridges throughout your career. It's always very helpful, because you never know, when you're going to go back onto your path, or when you're going to interact with people again. And I'd like to give it an example. For instance, when I became chief editor at Nature Medicine, the people who brought me into this role, or sort of actively recruited me in, were my competitors, who were editors at Nature, and who I had met in many scientific meetings and events, and eventually became friends and acquaintances, even though we were competing for the same papers, but sort of developing that relationship professionally, even though we were, for the most part, competing for the majority of the part, allowed me to be in their radar when an opportunity came up and they felt that would be beneficial for their journals to bring me over. So I think, cultivating these relationships with your friends and your competitors and everybody that you can, it really can help you in sort of moving your career forward. And my last tip is that I really think, and as I get older and more senior in what I do, I think this is even more important. I think that values are really important when you're choosing who you work for, because it is very difficult and very painful to work for an employer, whatever that might be, that you don't share values with. So because that's not going to change. I mean, you can change many things within your work environment, but sort of the philosophy of the people you work with, the things that they consider the most important, that those won't change because of you. So you have to have some alignment on your values and the values of your employee. And I just share the ones that, our own editorial values statement as Nature editors, which is something that I worked with other people to craft, but sort of represent how we place ourselves and the things that matter to us. We don't need to share the same values, but I think your own values need to be consonant with the values that your employer has, or the people you work for, or your institution, what they consider the most important things, because otherwise it just becomes unattainable in the long term, and it's futile to try to change your company's value or your institution's values, because they won't change because of a single person. So I think thinking actively whether your values are in agreement of the values of the place you would like to work, it will help you quite a bit with having like a career that makes you happy and fulfilled and satisfied at the end of the day. And I think that's where I'm going to stop, and hopefully have a few minutes for questions. I'm happy to answer anything you might want to know and that they have not mentioned. And if you want to reach out to me afterwards about editorial or anything else, feel free to do so. Here's my email. Thank you.
Thank you, João. It was really great, and for one that have known you since you were like just a little kid at Inca. It was very clear that you could do whatever you wanted and be successful doing it. But I don't think that anybody ever thought that you would be where you are, but again, very successful. So that part of the prophecy was always fulfilled. But we didn't expect you to end as chief editor in Nature Medicine. It was not something, very simple to achieve. So congratulations on this amazing career. And I was wondering if you ever felt, because of you always had all these many opportunities and many things that you could have done. Have you ever wasted any time thinking, what if? And if you could have done something else, would you be happier? I know you're very happy. At least on the Instagram feeds, you're very happy, and it's really a lot of fun, or the opera, or the cats. But do you do any of that? Is it something that people should dwell on and with the what ifs?
I think every once in a while, it is a good exercise. It's a good mental exercise, as long as you don't dwell on it for too long, right? Because if you start dwelling on it for too long, maybe that is a signal that's time to take a different path in your career, right? Because, first, I should say, people have different objectives in life, right? And they have different priorities. And I think the more you work with people and you manage and to lead large teams, you understand that people are motivated by different things and they need different things at different points of their lives. I don't think I dwell too much on of course, I imagine how things would have been had I taken a different path with my life, and they probably would have taken me to very different places, which, I would like to imagine that I would have been as successful as I am in the one I chose. But I don't think it goes beyond that, to be honest. I think I'm fortunate that I landed on a job in a place that sort of brings together the different pieces of expertise that come from different aspects of my training and my career. So in my current job, I use as much of my knowledge as a scientist as I do of my knowledge as a physician, and my ability to talk to physicians in their own language, which is a little bit different from the language that basic scientists might have. So I think in the end of the day, it kind of fulfills me to know that all these different steps of my career contributed to where I am now, and I can actively make use of the different skills that I've developed at different places with different people at different times to be successful at my job.
Right. And have you ever felt like boxing? It doesn't feel like it, because you have taken different career paths and found success in all of them. But I sometimes feel that the students here, they enter the undergrad programs and they only see one path going forward, masters and PhD and postdoc, and ending up as a clone of their supervisors. Have you ever felt like that? It was something that concerned you to make your one very particular way forward?
Yeah. I think maybe in retrospect of a little bit, I think all of us tend to model our careers on the people, well, as long as we like them, the people who trained us. And as long as we don't have too big of a regret of the choice we've made, it's unavoidable. Those are the people who are the most influential people in the development of your career. I mean, to be very honest, I do think that in Brazil, we tend to be a little too attached to sort of the traditions and I'd say the lineages we come from, let's put it this way. And that I think people would benefit of having a little bit of a broader exposure to what other people are doing, what other opportunities are out there. I think, in my own case, I went to a place, for instance, for my postdoc, which was the place where my PhD advisor went for a postdoc. I didn't work with the people that she worked with. I sort of followed a different path, but, I was in that same environment, who was also the same environment where her PhD advisor had gone to. So in some ways, I was part of that lineage. I think I was fortunate in the sense that my lab, for instance, was a very diverse lab and allowed me to be exposed to many different types of research and ways of doing research and people with different research backgrounds and interests. Had I gone to perhaps another lab where the exposure would have been narrower, I think I might not have developed my career the way it did. So I think in retrospect, maybe I would have made different choices if I knew, the breadth of science that I know now and the breadth of people and the wide network that I have now. But I think I always tell people, just look a little farther. I mean, I'm often asked to talk to early career researchers about careers and how to move on to the next step. And this is really the piece of advice I always give consistently. Just look a little farther from what you're looking now, because you're going to see a few different things that are not necessarily in your immediate radar, and they might change the way you're making your choices.
Joe, thank you. Thank you very much. I think on that topic, you mentioned the soft skills and how that can make the whole difference, even with two people who have the same opportunities, how far they can go. And from the career choices that you made and the changes, we can see that you are very determined. But it's not rare. And when I was in the US, I saw that happen, that when a person chooses to do something different from academia, at least at the time when we were kids, that happened a lot. They thought you were kind of failing, oh, you're giving up. But when we see you today, you see, it's easy to say, oh, he wasn't giving up. He was following his call, right? So I wonder, do you have any advice about that time point when you decide to make a change that is unusual, and people start for the better telling you not to, trying to change your decision? And what aspects did you hold yourself to when you decided, no, that's what I want to do. I want to go out, get out of this amazing lab in this amazing building and go towards a career that is seen as accessory, but without that, we don't get anything, right? Because it's publishing that is basically is today a measurement of how impactful your work is, and grants and positions are offered based on that mostly. So I wonder how, or what could you say about that strength, right? What could help having the strength to keep with the choice and the change? Because both you and Bernardo mentioned, which I think is very valid, change is necessary, right? In Brazil, it's not usual because most of the positions that we have in research are public positions where you have to do a concurso, right? A process through, and if you decide to change institutions, you need to give up sometimes your whole retirement plan, because if you change, you lose what you got when you got into that spot. Mm-hmm. And so, it's on that end. Would you have anything else that you could say?
Yeah, I think this strength comes from the mindset that, nothing is set in stone and nothing is forever. So there's no change. I mean, professional change, they cannot reverse and go in a different direction later. I think I realized that very early, also earlier than other people did. But I think that's the truth. You know, you can, especially when you're in the beginning of your career, I think that the more senior you get, the more big changes become more complicated because there's more at stake. You know, sometimes it's not just you. Sometimes you have family. You have to consider other things that are extenuous to your own, priorities, or it just becomes more complicated. But I think especially in the beginning of career, you can give yourself the room to try things and to move on in a different direction if that was not the right choice. And I think that's valid not only for independent positions, but even at postdocs. You know, I had friends who went on postdocs that were miserable, like miserable. And we would chat and talk, and it was like, why are you doing this? I mean, obviously nothing good will come out of it. I mean, honestly, there is no paper in Nature that's worth this level of suffering. There isn't. Nothing is. I mean, the only possible outcome is that you're going to publish this paper and you're going to quit science completely at the end. That's the only thing that can happen after this, right? So I think you just have to really, it's not a failure to change directions. I think you tend to think, maybe you tried something and you went that far and you realize that that's not the best for you for whatever reason. You're not failing if you take, an alternative path. I think you were failing if you continue to go in the path that has no end. I think that to me is a failure, because in the mid and the long term, that's what's going to matter. And I think having that awareness and having hopefully a network of people who can also give you sometimes the outside perspective on what's going on with you, it's helpful. I think in my own career trajectory, I think to one of the when I was really considering serious, I had gone far enough on the selection process to become an editor to the point that I had to tell my boss, I might actually be leaving tomorrow because I'm going to get a job offer. I think to me, I had a very honest conversation with him, and he, laid out all my qualities and the things I was not good at. And I remember very vividly that how he told me, you're absolutely smart, but you cannot focus on one thing for a long time. And it's like, yes, I cannot focus. So let me find something or a career path where that is an actual asset without being a setback, and I think that was, to me, really the deciding factor, when we had that conversation. And it was really clear to me that, I would never be happy doing the same thing for 10 years or 20 years, and that I needed to have breadth. And sort of looking back to my own development up to my postdoc, I think, in revisiting the decisions I had made earlier, that also became very clear. So from that point on, I really had no doubts.
Thank you. Thank you very much. I think there was one question on YouTube that was a little bit specific, but I think I can edit it a little bit. But just because you basically work with several with people with several skills. You mentioned that you need to work with the art editors, and you need to work with the different parts of the publishing as a whole. And I wonder if those, I guess the question was, these positions are also filled by scientists, or are they for instance, making the figures was the specific question. Are there scientists that end up getting those type of positions, or are there are specific paths for those accessory points, and basically the scientists would be better in those journals doing the editing itself because of this knowledge of the fields?
Yeah, it's sort of for instance, in the art, it's kind of a mixed bag. There are people who come into work with editing art for science. By art, I mean figures and making things look prettier and easier to understand and make sense of handwritten drafts that people send you in the back of the napkin of the dinner that they had last night. And some of them come really from an art perspective, and they are art designers who have an interest in science, for instance, and they land a job in a scientific journal. Other times there are people who come from sometimes a background in the biological sciences, but they always had an eye on design. So it's a little bit of a mix of the different paths. I don't think it is a requirement for some of these positions to have a scientific background. It does help. A lot of our editors have some scientific training, but not at the level, for instance, that the editors of the journals have. The editors are pretty much bench-trained scientists. I say 99.99% of them.
Mm-hmm, mm-hmm. Okay, thank you. And so thank you very much, João. Bernardo is still here. Gil, I think, just left, but especially John, who came in, has been here with us the whole time. And I hope that the students and everybody who was able to watch, Bernardo's there, and we're able to take things from this discussion. Definitely the emails, at least the emails, so you can email people who have gone through paths that might interest or decision making that might relate to the moment that you are. And definitely, if they accepted our invitation, they definitely have interest in seeing new minds going towards the right places and being able to improve research in all the fields that it might touch. Right. So thank you again, everybody, for being here. And I think I'll pass it to Bruno.
I just want to thank everybody that's participated, Emiliano as well. And but I also need to thank Miriam that's put together this session. It was a lot of work. We had some technical issues at the beginning, but we managed to fix them as well as was possible. Thank you guys again. Thank everybody that's stuck with us, either here and on YouTube, and see you guys around.